Uou. Agora sim, tá. Só para fazer uma breve introdução. Bom dia a todos. Agora nós teremos o simpósio satélite da GSK. Quem estará fazendo a apresentação será a Luciana Tarbes. Ela é farmacêutica, mestre e doutora na área de saúde pública e ela é gerente de Value Evidence da GSK Brasil, né? GSK Brasil, e ela então fará a apresentação do tema... o tema que está escrito. Desculpem. Então eu vou passar a palavra à Luciana. Obrigado pela presença, Luciana.
Obrigada, André. Tá funcionando. Acho que eu vou ficar aqui, talvez, porque isso não vai favorecer meu salto. Mas, o tema, na verdade, é caminhos e formas de fazer parceria entre a indústria farmacêutica e o setor público, e aí a gente vai trazer um pouco a experiência da GSK Brasil, como que a gente tem contribuído com isso dentro do país, tá? Esse é meu conflito de interesse, eu sou funcionária da GSK. Egressa da Fiocruz Minas, meu mestrado foi lá, então é um prazer estar de volta na Fiocruz.
Great to be back at Fiocruz to speak about science.
Bom, eu fiz aqui uma analogia.
I've made an analogy here of a plantation, which is a bit of the path of clinical development and research, the development of the drug until it is released at the market, what are the trials conducted, the possibilities for partnerships.
Então a gente fala, deixa eu ver se o pointer vai funcionar. Não funciona. A gente vai falar desde ciência básica.
We're going to speak about the basic science, how GSK invests and incentivates science in this area, phase 1, 2, 3, and 4, which are the clinical phases of the development of the medication. And we also can't forget about the observational studies, which is where this clinical trial part will be born, what population will be used, what is the incidence in the population, how prevalent is this, the burden that this disease brings or not. And I've made an analogy here. The plants are being watered. It would be the independent platform that GSK has, which is the fertilizer of all of this. So how we can fertilize the science and the studies in any of the phases of the production of this medication. So starting with the basic science and preclinical phase, what do we have here in Brazil? We have an institutional program by GSK, which is inside our global health umbrella, which is called Trust in Science. It's a partnership that GSK has. It's institutional, so we look for a CNPq or another entity to bring investment. We invest an amount of money and our partner invests another. So the researchers submit this in a research open call. They submit their projects, and that's when we start, after they are approved, we start the partnership. It's not a partnership that GSK simply gives the money. We evaluate the project, look at it closely. So Fiocruz has had some projects considered in the past. And today, we currently have some projects with FAPESP. We invested 10 million in 10 years, and FAPESP another 10 million. The interest is normally, this is a project for Latin America. We have our greatest investment here in Brazil, and our subject of interest In the open calls are usually neglected, diseases, immunoinflammations, and HIV. In Brazil, we considered these more because of the demand. It was an open call. We looked at neglected diseases, leishmaniosis, vaccinations, and usually in Latin America, immunoinflammation. This project started in Brazil in 2012, and we've had about 24 million invested by GSK. When we go on within this umbrella of the Trust in Science and Global Health, we are expanding our partnerships. So beyond the financial partnership and evaluating projects by GSK Global, this is our global investigator that helps us with the Trust in Science program. This is an article that was out in Nature. And we also signed a partnership between FAPESP and Biominas to support projects that are startups, because we came to the conclusion that us researchers know very well how to do science, but we don't know how to sell our projects to the industry. So how can we make this partnership happen? GSK entered in the technical area. How can we look at the investigators in the basic area, help them technically, if this project is interrelational, if it's following the technical part correctly? And Biominas supports the business area, how to sell this project, how to show it to the market and other pharmaceuticals. And finally, at the end of last year, we signed a partnership between FAPESP and the British Embassy to bring 2 Nobel Prize winners. We don't have the date defined yet because they have a very busy schedule, but there will be a great event to incentivize, to understand what their path was to win the Nobel Prize in Science, what was the path they made in this research. Now, going back to our plantation, when we get into the clinical phase, we're financing a lot of basic and preclinical elements, discovery of alpha therapeutical molecules. Then we go on to the clinical part. And we can see in the trials in Brazil, we conducted research. The normal standard in the Brazilian environment is phase 1, phase 2, and phase 3. These are normal in the area of oncology and HIV. Phase 2, phase 1 less normal. A lot of investment in phase 3. And phase 4, you can see a progressive increase, progressive observational increase here. What we observe in GSK is that this model repeats itself. We have high investment here in Brazil in phase 3, which is very good because we already offer this access to medication to the population. We test the medication on the Brazilian population. We engage the researchers in global projects, and usually these projects are multicentric. I think most of you are familiar with this. They are multicentric, so there is a global protocol with, you know, what the kinds of patients are, the characteristics that will be included, and Brazil is included as a center in which this protocol, we translate the protocol and enter this into this study or trial. For these kinds of trials in 2017, we had more than 150 research centers and units involved in our trials, more than 150 Brazilian researchers involved in our investigations, and more than 2,100 subjects or patients recruited in these trials. The investment in Brazil in 2017 was around 30 million reais, and globally in the year of 2017, 18.5 billion reals. when we go on to the observational studies, and this is very particular here in this country because we're speaking of how the system works, how the population is distributed, how this disease or illness happens in your country, we have more autonomy relating to the protocol because it's what we need, what the ministry needs. So what are our demands? So we can proactively submit protocols to the global team. So we compete with the whole world in order to get our financing. It's like a foundation that supports our research, and through this we can design the protocol, make suggestions, say why we need this, and start the trials. So most of the time these are the models, they are transversal. We do a lot of transversal work, a lot of descriptive ones, trials for the use of medication, and also studies relating to the costs in health, because this is all very specific to our country. The areas in which we are most interested in are the respiratory area, lupus, vaccinations, and oncology is green here because GSK, Warren G of GSK, the RNG of GSK is changing its route a bit. So we sold the part of medications that we had already launched in 2014. For oncology, we continued studying this oncology pipeline, and now is the moment to relaunch molecules. We are investing greatly in this oncology area, so most likely we will anticipate and start observational studies in the oncology area. These numbers are for 2015, 2017. We had more than 40 publications. So this is a bit of a smaller structure in Brazil, more than 40 international and national publication with Brazilian data, and about 30 million reals invested in this kind of study. And then we have this part of watering the plants. It's a platform that GSK has launched. It's a new platform format. As I said previously, it can be in any of these phases of the clinical development. Today, previously, well, this platform always existed, but I will speak more of this later. We do more of a certain kind of proposal in these areas. We speak about asthma, lupus, HIV, oncology. What interests us in these areas? So the researcher can submit to this platform. Once they submit, this goes through a national evaluation board. Once it passes this board, it goes to the global board. The global board evaluates the study. If it is approved, the person receives the financing, or it might not be money necessarily. It could be a medication. It could be a support for this publication. It doesn't have to be the whole project. So this platform is a lot more flexible. We have 9 approved studies last year and this year. We have them in HIV, asthma, and lupus. Lupus in the immunology area. This was our pilot area at GSK, and that is why we have more approved studies in this area. However, currently we have launched this platform that is open. So you have, it's the supported studies program by GSK, and you can find here, these are the areas. You can click on each area and you will have the related subjects there. So we are really connected to the global team, so the material has to be in English. It has to have the quality, a global quality of science, because we are competing with the whole world. But fortunately we have had great results. From 3 that were submitted for lupus, 2 were approved, and in the last open call of 7, 4 were approved. So this is good recognition, and this is a way of obtaining the financing. The advantage of this platform is that GSK cannot interfere with the study. We are not the authors of this study. We cannot interfere with the method. We can make questions. So as the financer, just as FAP, you can do this. You can say, I don't understand what's going on here, but we don't interfere in the objective of the study. And it is not necessarily connected to our products today. This is a platform that will have a partnership with our pipeline that I will show. So it's a way for Brazil to get ahead in the pipelines and to build this future with GSK. So inside our pipeline, here we have great investment in oncology, HIV, immunoinflammation, respiratory, other pharmaceuticals, which is a minor part, and vaccines. Here I brought just a brief summary of what we have in terms of volume of pipeline and assets. So in oncology we have 16, being that most are in phase 2. In oncology we have a number of molecules that are approved in phase 2. So this is phase 2, HIV, 11 assets, 3 that are being launched, 4 in phase 2. In immunoinflammation, it's more distributed, so we have lots of things in phase 1 that will still grow, and 4 in phase 2. And in respiratory,
we have 10 assets, 10 molecules being studied, and here are the 4. And in vaccines, we have 17 molecules under study, but the majority is in phase 2. And what I brought here, that probably is not a very well slide for you to see, but this content is easily found in Google. That within oncology, our priority in short term, let's say, is multiple myeloma and head and neck cancer. That's what I would like to call your attention. And in terms of vaccines, we have approval in adults. And now we are having studies in children for Becerro, and the essential virus, that is a platform that is of great interest, and we include children and adult populations and also mothers. And that's it. Well, our mission is to invest in science, and we've been doing that for 110 years and building science in Brazil. Here I focused in R&D, so we have a little bit of strategies, which is not the focus here, but we have an innovation program launched in SJK, in which we created this program called Together, and we opened partnership not necessarily in R&D, but with the 10 challenges for startups. And yesterday we launched another program to generate data in this innovation umbrella of GSK, and we called Data Toll. This is a hackathon of data tools in terms of epidemiologies, and I think it also links with the Innovation Hub, and we are learning, and our IT director is here. She is responsible for the innovation program. So that's it. Our president is here, and I hope we have 110 more years contributing and investing in science in Brazil. Thank you.
Alguém tem alguma questão?
Do you have any questions, any comments to say to her?
Comentário?
Anything to say?
Bom dia. Eu só queria
I would just like to know a little bit more, because you talked about vaccines and products and some diseases that may be neglected, but are they all focused on biologicals, or you also have synthetic molecules?
Temos dois. provavelmente distribuído de forma
Well, we
diferente
have
entre as áreas.
both, probably distributed differently. In oncology, we'll have a lot of immune and biologicals, but in respiratory, we'll have synthetics.
Está em transmissão.
Bom dia. Geralmente o projeto para
Good morning. Generally, the project to be approved and evaluated, how long does it take for you to evaluate the project? The project that the person submit.
Aqui, o André resolveu. 98.
Well,
Bom, vamos lá.
let's see.
Transparência.
Transparency for pharma,
a GSK é muito
because
grande,
GSK
então
is
são
quite
estruturas um pouco
huge, so we
diferentes.
have different structures.
Pharma, que a gente chama,
for
que é a
pharma,
parte de
as we
oncologia,
call, that is the
tirando
part
vacinas
of
e HIV,
oncology, except
pharma,
vaccines and
98
HIV.
dias. Você tem que
So
ter o
pharma, 98 days to have the result.
vacinas está seguindo
For
o fluxo,
vaccines,
mas eu
you
acho que ainda não
follow
foi estabelecido
the
prazo.
flow, so there is no
E VIV é muito,
deadline
VIV é até
established.
mais rápido. Desculpa,
And
HIV é até mais rápido.
is much faster.
Geralmente é mais rápido.
Generally, it's
98 dias.
faster than 98 days. But if it is in the case of basic research, the person will submit the project, and we don't know if the project will launch or not. So how do you evaluate that? You send to a person to understand the viability of the project, or if there is a risk?
Avaliantes.
Well, we evaluate it before. As it is basic, we don't have technical knowledge in Brazil, so it will be evaluated globally, the viability and interest. If it is via the Trust in Size project, it will be between FAPESP, for example, and JSK altogether. So generally, they invite ad hoc researchers to be part of the evaluating team. Any other questions, comments? No. So, Luciana, thank you for your presentation, and thank you. And right afterwards, we'll have another satellite symposium.
Isso, Você quer que eu apresente?
Você quer adiantar?
Mas depois, na hora do... Só para convidá-los para...
Ah, sim. Deixa eu ver, o próximo é 10, né?
É Patrick Gertin, da GE, falando sobre inovações em vacinas e produção de viral de vetores.
Acho que a gente pode dar esse intervalinho.
Isso.
Teremos pessoal, bom dia, teremos breve intervalo.
Well, we'll have a small break, so we come back at 10 with a GE session. With Patrick Gertin, who will make his presentation on new technologies for vaccines. Thank you for being here.
Depois que o botão cai,
obrigado, Luciana. Muito bom.
Obrigada, André.
Desculpa qualquer coisa.
Não, mas...
É que tá um pouco corrido aqui, que a gente tem também um limite de cadeiras na sala do museu, não deixa a gente deixar em pé. Aí tem que ficar vai e volta, controla. Tá um pouquinho
But I think that's why we need to continue to move forward on different therapies, gene therapy, cell therapy, viral vectors, you know, in anticipation of what could come next. Because just like, you know, any form of nature, right, the evolution and adaptability, you know, we're evolving all the time, and the diseases that are challenging us are evolving over time. So I think that looking at these new mechanisms, you know, and how we use them, you know, how do we use a specific delivery system? I mean, if you look at some of these features on the right, components on the right, you know, they're really elegant mechanisms on how they can act with your body's cells. And so looking, you know, just on the viral vector specifically, looking at from a 2016 study, looking at some of the numbers in the clinical phases, you know, we look at a total of, you know, 726 at that time, and proportionately how they're represented, the largest percentage of those being the adenovirus, you know, followed by the retroviruses, so on and so forth. But you can see the relative breakdown. And so, for example, if you look at all the products, both therapeutic and viral, in the clinical phases, you know, whether it's, you know, phase 1, phase 2, phase 3, what have you, you can see over time, since this period and even more recent studies, that percentage is increasing. So the total number tends to increase overall in terms of a trend, but the proportion of these respective components also likewise increases. So, you know, if we look at vaccine production today or in the recent past, we look at, you know, processes that were really developed decades ago with older cell substrates or eggs, limited purification. You know, they were processes difficult to scale up with certain laborious steps involved in the processing. You know, unfavorable process economics, some of the increased regulatory requirements. And so, again, if we look at each of the components within these categories, we see that, you know, the egg-based production or other cell types are being replaced by plasma cell or more regulatorily accepted. So when you go for your filing, you know, you have an easier path to it because you're using a well-characterized cell line that has a very rich history. So again, this is a critical component. You know, where before it was very expert driven, you know, now there's efficient purifications based on chromatography. You know, again, science and art kind of coming together, so to speak. So the scalable technologies and the single-use technologies are certainly playing a significant role. And, you know, I can tell you, and not to digress from the point at hand, but with respect to some of the single-use technologies, I've used those for microbial fermentation, mammalian production, you know, subunit vaccines, virus-like particles, traditional viruses, vaccines, et cetera. So again, the point is, you know, one should try to use technology that's adaptable. Again, as I said, for what we have for today, but what we anticipate for tomorrow. And then with respect to the unfavorable process economics, you know, efficient and rational process design, we'll talk about it more in a moment. You know, the QBD, or quality by design, continues to be quite essential for the rapid approval and forward movement of any process. And really that starts from the beginning, right? You know, so before you embark on a particular project, you know, use the tools that are available, use the literature that's available, you know, talk with people, team up with other collaborators who have knowledge in the space, you know, and really take value, you know, contribute to them and accept help from them. So, you know, closed handling, closed processing, as you know, always the environmental considerations are always a challenge. Closed processing, especially in the recombinant protein space, has really moved forward. Again, that's what allows a lot of ballroom settings rather than traditional room-by-room segregation. So things like connectivity, closed processing, significant filtration applications, these are things we should think about. And then, you know, back to the quality by design. I mean, at the end of the day, you know, all these things, you know, it's tremendous knowledge, but really we know what target titer, what volume, what product per time we need to produce. We also know what are the critical process parameters by which we have to operate our machinery, and ultimately what is the critical quality attribute, CQA, of the molecule and some of the contaminants, you know, things like clearance of host cell protein, host cell DNA, these types of things. And, you know, where there are still many processes with serum, I think they're becoming less and less over time. We see the chemically defined media. So, you know, again, this is a reiteration of the earlier slide. I won't belabor the point, but one of the a single-use platform utilization, this happens to be our virus production, you know, moving from left to right. If you look at the molecule design, the cell design, selection, cell culture, you can see that, you know, it really starts with the cells and the constructs and the bioreactors, the clarification, the downstream considerations, that space, and, you know, how efficient that process for your molecule. And we always like to think of our molecules in particular, you know, from a very important perspective, right? That's our company's business, that's our company's, our future. But, you know, again, talking about the earlier point about flexibility, you know, there may be a point when you want to use certain machinery to produce analytical components for the testing of your molecule. So again, if you wanted to produce a series of antibodies for assays and do that in-house, you know, if you had the equipment and the right know-how, you know, that can certainly benefit you. And so, you know, taking a look at some of the single use in the vaccine production, you know, oftentimes manufactured in small batches. So, you know, in that way, single use is appropriate. You know, the campaign manufacturing is important. And again, by no means am I anti-stainless steel. You know, again, I have 30 years in the industry, 16 of which was in stainless steel and glass, 14 of which is in single-use technologies. And I can tell you that each one of them has their place in their application. You know, it's really how do you choose to use it and what's right for your process. You know, and I've always said as well, you know, if you've already invested in a lot of technology, you know, don't necessarily abandon it, but learn how you can hybridize or modify and make more flexible the different unit operations. You know, the third bullet point is a really important one because of the pandemic preparedness. We did a contract a number of years ago. I can't talk too much about the details, but again, it was in pandemic response, and it was about generating, you know, 30,000 doses in less than a 6-month period, starting with nucleic acids. So again, it's really important to address any of the market needs, whether it's pandemic response, whether it's endemic or what have you. You know, some of the high cost constraints, you know, with vaccine, you know, need to have process improvements. And if it's in a single unit operation, that's still going to bring you a lot of value. I think a lot of people philosophically think of things, and listen, you know, we're only human, right? So we think of things in an all or nothing fashion, but it doesn't have to be an all or nothing fashion. Okay, so, you know, as I've always said, you know, just to keep the mind open and see, you know, what are other people thinking, the industry doing, and what are my needs? And safety in the cross-contamination is an obvious one. So the single-use processing, the closed systems, you have a single-use bioreactor on the left, single-use connectors, and the ready-to-process column. And again, one of these considerations, you know, really, again, is the, you know, negation of certain infrastructure, clean steam generator, WFI still, you know, these types of things take tremendous energy and money to maintain. So again, taking a look at some of the compiled data from a 5-year period previously, substantial savings with single-use technology. We see the first column. I won't belabor this point, but really, you look at each of the respective categories, and with the single-use, consumables is always going to be your largest cost. The capital facility cost is going to be lower. The labor is generally going to be lower because, again, with some of the closed processing of single-use ops, operators can do dual roles, don't have to be necessarily segregated to a single role. Is my sound okay? Am I okay? Yeah, okay. Sorry, I can't tell really from my perspective. But, one of the most significant ones, and this is more of an environmental consideration, but I think we do have to be environmentally conscious while we're doing this manufacturing, is, water utilization. The water and energy utilization in our analysis over time has really relates to about 75 to 85% less water use. And a lot of that comes from the absence of CIP or SIP for respective unit operations. But the main point is, as water becomes more and more precious, I think that if we can achieve our goals for manufacturing in an environmentally conscious way, not only is it a cost savings, but I think it's a philosophical advancement. So but the bottom really just shows some of the unit ops in terms of mixers, skids, reactors, and purification skids as well. So for the adenovirus vaccine production, just a schematic showing the scale up from left to right, what's entailed in the downstream. And on the right, really is an important component of it is really the analysis, okay, because we have the steps for the unit operations, but, a key variable is the quality metrics and what testing's going on throughout. And again, what's the timing of that testing? Can you do real-time analysis, or are you going to be delayed? So, the cell substrates for vaccines, if we look at the production system on the left, really showing more of a traditional cell platform, on the right, more bacterial, showing the vaccine types, whether it's attenuated, inactivated, SUV, VLP, so on and so forth. On the right for the bacteria, the conjugated polysaccharides and things that, again, can oftentimes have challenges from a downstream perspective. But the main point is that it's really a broad spectrum that we have to choose from. And as you're looking at the cells, things like, again, what's the history of the cells? What are the characteristics of the cells? Do you get a higher titer from a particular cell strain that happens to be attached? So you have a little extra challenge for the attachment, but you gain the benefit from the overall titer. So, the cell substrate, a lot of the ones nowadays that are really preferred are, the Viros, the MDCK, the duck cells, so on and so forth, good safety track record, animal origin free, good virus propagation. The media available, again, I think it's important to keep it animal component free wherever possible. And looking at, the adherent characteristic, as I mentioned earlier, versus suspension cells, cells like, Viro, for example, are attachment dependent. For example, HEKs can be attachment or suspended or suspension. So I think it's important to kind of balance, what works best for you and really what cell line results in the best quality attributes for your virus or vaccine. The feature about the attached cell lines as you embark on those is, it is a more complex scale up. But again, if you're getting, 10x tighter, then that's value. That's worthwhile. So looking at some of the downstream components, in full disclosure, most of my career I've been upstream, bioreactors, et cetera. I have done some downstream. I don't tell my downstream colleagues that I've done that because they go, really? But I have done enough to know a little bit, so sharing some of this. But again, with the clarification, first step, filtration, normal flow filtration, TFF, centrifugation. If you look at centrifugation, I mean, there are some single-use centrifuges nowadays, but, most people, for the economics and the relative centrifugal force that you need, will go with a traditional, but again, that encompasses cleaning. Okay, so you have the capital cost of the unit, and you also have the CIP. Looking at some primary capture, focusing really on the fourth point down, the chromatography, IEX, mixed modal, AC, affinity chromatography, hydrophobic interaction, so on and so forth. There's also the last bullet, the membrane platform, which can also be very effective. So when you think of these things, you need a particular step. Again, whichever step, whether it's starting out with an affinity step, followed by an anion exchange, followed by cation exchange, etc. But it really comes down to the other decision. Again, how are you utilizing that resin? What's the resin lifetime? Are you using a traditional glass and stainless steel column? A bit about that. Excuse me. We'll talk a little bit about that in a moment. About some of the labor costs associated with packing a column. And also some of the qualifications of those columns where, in certain instances, you can put that burden on the vendor or manufacturer of that unit operation. So, with some of the core bead technology available now, again, essentially, this is a modern alternative to SEC. And really what it is with the technology, it's an agarose bead, but rather than just having certain divots or compressions in the resin or surface ligands. It's really channels through the beads with the ligands attached on the interior channels of the beads. And what this actually does is this increases the speed and efficiency and increases your total surface area that you're able to use. As you can see schematically, we have the virus molecule, the largest component on here, the fragmented DNA, the host cell proteins. So again, this would capture the contaminants, allow the and so with the core bead application, there's also, especially for some of the viral work currently, Capto-Devirus, which is actually another agarose core type of bead, but with the sulfate-like ligand on the interior channels. So the point is that this change to some of the resin design has actually enabled the increase in efficiency and speed. So, again, is it right for your molecule? I think you really have to test it and see. So, taking a look at the applications for some of the core beads, you can see a pretty broad spectrum of molecules, virus, et cetera, that can be applied here, even VLPs. I think there are some commonalities among all of these components, but again, there's also unique differentiators and also challenges to reduce your contaminants, whether they're product-related or non-product related. So, a cost breakdown, some of the steps in the simulation alternatives, looking at the literature search. Again, as I said earlier, check out the literature, see what's being done out there currently. Talk to your colleagues within your team what they're doing. There's also, with the process modeling from a very powerful tool. Set up different process alternatives, and then evaluate results and identify the economically feasible unit ops. And as I said earlier, not taking an all or none philosophy, where does a particular, you might get from the replacement of one single unit operation, you might get a huge benefit. Okay, it might save a day in processing, and if you get X number of batches a year and you save a day in processing, you might get an extra 1, 2, 3 batches per year, and again, that's really significant. And then, again, process development, it has to be done. You can do all of the other things in terms of modeling and literature searches and communication, but it really comes down to the process development, to really evaluate these components. So if looking at some of the process alternatives, we look at 8 different scenarios here, starting on the left. upstream process, detergent treatment, upper portion of the bracket, nuclease treatment, or lower the clarification with NFF, TFF. And as you can see with all of them, I won't bother reading. I won't bother reading each of the respective. Sorry about that. That's actually my alarm. It's not my phone. No, I wouldn't allow the phone. The alarm, yes, no phone. But what we see here really is a comparison of 8 different process flows for the same molecule, for example. And really, again, I'm going to show momentarily some of the cost per gram to produce and apply this this model or this evaluation. But again, it comes back to the primary component that I described earlier. How is that for your molecule? How is that for your process? What are your acceptability specs? The quantity of DNA or quantity of host cell protein per dosage or per mg. Okay, so you might get a process that is apparently economic according to a modeling exercise or according to one molecule's evaluation. But again, if that's for molecule A, if you apply this to molecule B, it might be a little different, right? So again, everything, some things that might cost less comparatively, you might not still be the top performer. So again, you may have more challenges at lower cost. Okay, so again, it's always important to look at this. And if we look at the cost per dose of the different bioreactor scales, this is a schematic representation of the cost per dose on that last exercise that I just showed. And as we can see that, comparing one to the other, and one of the main features of this slide, okay, there are subtle differences between each one, but I think from back to front, looking at 50 liter to 200 liter to 500 liter, I think what you realize, and a lot of people know this quantitatively, and a lot of people know this intuitively, you get a economy of scale, okay? Because of some of the materials and some of the operational logistics, again, you really are gaining financially from the scale up. And then, looking at some of the contributing cost factors for each of those respective models. If we look at it, for the royal blue color, the capital, a lot of times, you'll get a higher cost if you have something like, the certain stainless steel resins or housings, depending on the cost of those housings. You have components like materials, obviously represented by the dark gray, and those tend to be largely equivalent. The consumables cost, again, where you'll get that's the light blue color in the center, and really where you'll see that for the consumables cost is if you have things like membranes, for example, again, because that's representing, something that's being utilized, as a consumable. The labor, if you have a traditional column, you're going to have to pack that. You're going to have to qualify that. So if you look at the darker portion, the dark navy blue color, again, the labor for packing, qualification, that's just sort of a very simple example. And then there are other extraneous costs. But if you look at the total, sort of varying, US dollars per batch, varying, somewhere between about $55 and about $85. So that's about a 35 per batch difference. But, again, if you take any one of these steps, if we look back at, here, if you were able to replace one component in that flow chart, again, it could result in significant benefit. And then just the evaluation of productivity. Really, the core technology, this is just an up-close look at the core technology replacing an SEC step. And again, not necessarily replacing every single step, but just for the SEC step, and showing that with the respective matrix and the channeling and the ligand attachment to the interior channels of the beads, you can certainly gain benefit. And, just showing, and this is intuitive to most here, the principles of SEC with the higher molecular weight components being excluded from the pores, coming through first, and then proportionately according to size and hold-up time through the system. And the productivity, if we look at the, again, economy of scale or product per time, we see that the core bead really, especially at the 2,000 liter scale compared to the 200 liter scale, certainly outperforms. And then if you see on the left, the core bead technology in a stainless steel column, again, certainly increases above the traditional SEC and stainless steel column, but still slightly behind the single use. And then, in conclusion, the paradigm shift for vaccine production from lab bench process to rational design, incorporating process economics, combination of single use and traditional. I think those are really good things. It goes back to our original discussion, right, about staying open-minded and potential hybridization and what works for a particular unit operation. And then, again, looking at, core bead technology, it may or may not be right for your process. We've seen it in most cases that it is absolutely right for a lot of processes. But just in the case of, for SEC, how does that apply itself? And then I would like to thank the team shown here, especially Mats Lundgren, who was significant in putting this presentation together, the Uppsala R&D team, who are terrific. Again, the Marlborough, Massachusetts, just outside of Boston team is very good at the vaccines as well, but for purposes of a lot of this study information today, the Uppsala team. And again, that goes back to one of the things I said earlier about the different teams. No matter where you're at geographically worldwide, bond with your team members, try to find out what they're doing, try to support them, try to get support from them, and just really, embrace the collaboration. So thank you very much for your time.
Vocês têm alguma pergunta? Do you have any questions for Mr. Dr. Patrick? Any questions? Alguma pergunta? Algum comentário? Não? Nenhuma pergunta, nenhum comentário? Certeza? Então, mais uma vez agradecendo a palestra do Dr. Patrick. Obrigado. Thank you very much for your presentation. E nós teremos em seguida outra sessão, então faremos um breve intervalo e em seguida teremos outra sessão. Thank you very much. Obrigado. Ok, thank you. Obrigado pela presença, pessoal.
Sessão que a gente pensou para discutir inovação no âmbito de biomanguinhos, e teremos hoje as apresentações dedicadas já à apresentação dos ativos de biomanguinhos especialmente, né? Ontem a gente teve a abertura com a apresentação da cadeia de inovação da Fiocruz e de bio, e hoje a gente vai começar a falar especificamente de biomanguinhos. Teremos então a abertura com a apresentação da vice-diretoria de Desenvolvimento Tecnológico de Biomanguinhos, que vai ser feita pelo Felipe Luca. E, em seguida, temos as apresentações do gerente do Laboratório de Experimentação Animal de Biomanguinhos, Rodrigo Miller, e depois, em seguida, a apresentação do gerente do Laboratório de Neurovirulência do Dr. Renato Marchevski, de Biomanguinhos também. Agradeço. E passo a palavra, Felipe, por favor.
Bom dia, bom dia. Vocês estão me ouvindo direitinho?
I am Felipe De Luca. I work with Sachiris and all of the rest of the team of the Vice Directory of Technological Development. You're not hearing me? Okay, I think it's working now. So because Sotiris could not be here and Marcia couldn't be here as well, so they asked me to make this presentation. It's quite short. We're going to speak about drug discovery platforms, but actually we're going to take a step back and say why we're thinking about platforms and services. That's the idea of the presentation. Let me try this out. Okay, so the first thing that we should consider in this context is the regulational conditions that we used to have relating to innovations in Fiocruz and Biomanguinhos. This changed recently because of the technology and science milestone, and then in 2018, based on this milestone, Fiocruz launched the innovation policy, and after this, the Fiocruz units, including Biomanguinhos, started thinking about what business models they could use, which would be possible considering the internal structure and the know-how that we have. So that's basically it. This was the alteration in the regulational environment that happened and motivated this presentation. So the first idea, considering this milestone in the regulation and this change in the environment, was to think about what our capacities would be, what are our positive characteristics in Biomanguinhos, and what we could use. So VDTEC, which is the Vice Directory of Technological Development, other tech labs, micromolecules, bacterial, biosafety. So there is a group of labs that are part of this Vice Directory. And along these years, they had the opportunity to work with or make partnerships with great pharmaceutical companies like GSK, and this experience is very positive. It's an advantage on our end. We can use this, not just the knowledge that is shared during these collaborations, but also the mindset of these companies was something important. Something that made these partnerships important. And this is something we considered and we are currently considering. Let me just go back here. Okay. So that's it. We have our internal structure with great experience, great thinkers and researchers here, highly qualified people in here with a lot of experience in partnerships with big companies for producing and developing new pharmaceutical products. And so there's a piece missing here. What does the market need? That's basically the project, this initiative of platforms and services that the Vice Directory wants to get to and wants to offer at some point. We will count on these steps. First, we need to conduct an analysis on the market. So the idea is to work from business to business. And so to collaborate with other companies and convince other businesses to pass their innovation cycle through the Biomanguinhos structure. And so we need to know what they need. The first idea is to analyze the market, assess the market. And then, considering the experience and the know-how that we have, look at this and compare what the market needs with what we have. So where we are and where we want to get to. That's the idea, and along with this we will identify gaps. The idea is to conduct an internal reorganization to eliminate these gaps or at least reduce them. And with this we can try to find business models or services or platforms that can attend to this initial demand by the market. It's pretty simple. Basically, this is something we started working and it's what we're doing. If you take a look here, it's all very recent. This happened in 2016. After that, Fiocruz started thinking about how they could work with this regulatory milestone. Then October 2018. So this is still in a preliminary phase. We have a structure of what we would like to do and an idea of how we want to do this, and we're walking in this direction. So looking at the vision that we have today, which is still very preliminary and superficial, we can already see some values or advantages that competitive advantages that could work in the future. The production of eukaryotic cells, viral production, the development and production of monoclonal antibodies, the recombinant proteins in E. coli, the sequencing of cultures, the increase of scale in bacterial culture, and even consulting for biosafety. So this is what we have today, but it's still very preliminary analysis. This is the future. So from this market analysis, we need to look at everything that can transform into a competitive advantage, and we need to make an effort to diminish these gaps and then launch these platforms, one or more service platforms that can serve this demand from the market and be able to create partnerships from this and help other companies to deliver products that are innovative and can contribute to our objectives, Fiocruz's objectives, public health objectives in Brazil. So that's it. Thanks. I told you it was going to be short. Thank you. Thank you, Felipe. So let's leave all of the questions for the end, okay? So Rodrigo Miller. So I'm just going to get started. Good morning, everyone. Can you see me? Okay, great. So we want to present the technological services that we can deliver today. What are our portfolios, services that we offer? Today we have internal clients, especially EVDTech. We have experimentation that already offers internal services. for all of it, DETEC, LATEV, and others who are also here. So we have cutting-edge technology, and we have our technological partners that we have used for preclinical trials for products, and in these partners are GSK. We have a huge infrastructure. I'm not going to get into all of the details, but the operational area is almost 1,000 meters. We have a special air conditioning system for the animals. We know that trials with animals today are very important in their specificity, and the data for reproducibility, it should be considered this so that the study can go on. So we have a team that is highly trained. They are certified. Internal certification has been created so that we know everyone is trained and specialized in what they do. There are not people who go into this. We have the internal control. We have 11 technicians, administrators, and all of these people in the lab. We work with rats, rabbits, and guinea pigs. We have activities for quality control and things that our control department utilizes. We have technological development, as Felipe mentioned, some areas that are providing external support, and technological upgrading and transference for the unified health system. We work with good laboratory practices. This is a frequent qualification for our professionals. Training in biosafety, consulting and infrastructure. For other locations, also inside Fiocruz, we have this consulting for structures and the experimental refinement for our clients. If somebody seeks us out, we conduct this consulting to improve the studies, the trials with animals, so that we can obtain a good result. And this is something that is very relevant today. The quality control, we have non-specific toxicity tests, pyrogen tests, stability tests, potency tests, virulence tests, and reactivity tests, skin reactivity tests. These are all tests we conduct for quality control. Also, the production of anti-ovalbumin in rabbits. We have technological development, lots of activities here relating to recombinant protein, quantification, bacterial quantification in animals, passive immunization, evaluation of vaccines. Today we know there are many difficulties to find the best vaccines and get the best responses. The antibodies, murine antibodies, production of monoclonal antibodies as well. And the methodologies that we use, there are a variety of experimental techniques. We do a lot of things and are constantly improving our techniques, and we are always in contact with our researchers in order to improve these experimental results and make them efficient. So with the mice, there are different pathways. We have cardiac puncture, saliva, etc. In the guinea pigs, we also have blood collection by cardiac puncture, and the rabbits as well. And we also work with chicks for the production for the blood collection. So there are a variety of items. I can't speak of everything right now.
We also have subcutaneous inoculation, endemic, intranasal, intrabrain, in neonatal individuals for collection as well, in guinea pigs as well, and also in rabbits.
de 2014 a
So
2018,
from 2014 to 18, we had almost 300,000 experimental tests. So we have repeatability that and every employee coming and being trained so that we can reduce the number of animals because we can have a specific technician working for that, and we know each one of them and their individual performance and how they work in the animal tests, and they have to be the only ones in the room. We generally have only 2 people in the room due to odor and noise and so on, so that experimental test and the responsibility of people there are quite huge so that the test is effective. We know that from preclinical to clinical trials, it's a difficult process, so in our lab we have to make it as perfect as possible so that we can evolve. Now we have 15 immunobiologicals under development in our lab. Some of them are really there. Every year we have about 700 trainings in the lab with our employees, and we have 12 there in the animal areas, and we have 700 trainings for them every year, and this never ends, it's cyclic. And we have qualification programs because it's not only people, we also have equipment, and we need to check the areas and the scales, because if we weight different grams, we have a different result. We need to be everything to be proportional, so we calibrate every equipment, and people, even their conditioners are checked so that the animal experiment may be effective. So that's what I wanted to tell you. Thank you.
Chamo então para sua apresentação, Dr.
Now I invite Dr. Renato Machewski for his presentation. Well, good morning. I'm sorry some of you are standing. There is a seat only for a short time. You may sit here. Anyway, at first, in honor of 2 architects, Luiz Moraes Jr., who created Biomanguinhos within a unit that you know is important, but secular Fiocruz is quite important, and the architecture of Luiz Moraes, and here Santiago Calatrava, who built the building that he says everything is curved, and I was somehow afraid of entering here, and obviously, you know, it's a pleasure to be invited to be here in the Museum of Tomorrow. First, I would like to congratulate my colleague Rodrigo Miller, and you can see he is quite thin, and it shows the non-stop activities he does. Laneu, and I'm part of this group. It started in 1982, and I have only 58 protocols with non-human primates, so you see it's a huge difference in the amount of Rodrigo has shown, and you can see that working with 58 protocols, it's a lifelong experience. So I would say that ideally, no monkey or no non-human primate should be used in research, but we have no alternative. We are obliged to use them. But our only idea is to have scientific development with the use of non-human primates. So I won't say I refuse using them, but in Dr. Akira's dreams, within this time I would be in Manguinhos, non-human primate would be used anymore in the end of my career. Unfortunately, it's not a reality.
Isso é para relaxar.
This is just a quick time for you to relax, because I see something incredible. I don't see any glass of water for a hypertensive and diabetic person. I can see a bottle of water there, but I don't want to take your water. No, please, take your water. But you are responsible for the invitation.
Então vamos ver se eu aprendi. Não é que eu aprendi? Bom, vocês estão vendo.
Well, as you can see, we had the first problem here when our colleague said that they were managers in the areas. I believe they should check that if Vivian and the reviewers would make this kind of verification. But 1 month later, nobody corrected anything, and we are receiving our material in the last minute. So what do we talk about animals? That they shouldn't be used. You all understood the message. The second message is that without them, we wouldn't have all the developments we have. They are unavoidable. So when we say we use non-human primates, at least in Fiocruz by EOCD since 1909 with Carlos Chagas, we understand that we cannot do anything in the medical area without them. So the idea is that monkeys couldn't, in fact, not be used. And the proof of that is that every year, and I have data from 2018, 100,000 non-human primates are still used worldwide, showing that despite all the ethical part involved in it, we still have this kind of work. So we bring this idea that we have to use them unavoidably because all development depends on the use of non-human primates. Despite Rodrigo being the owner of a lab using rodents in an incredible number, and he didn't mention this number, but you would be shocked on that, we still have today only 0.5% of researches on animals with non-human primates. If you take a look at these numbers, you will at least believe it's just a few, but 100,000 in the world is still the guideline, and this progress cannot be prevented. Another very important part of our history is understanding something called divergence. When we use, for example, a chimpanzee or a bonobo, that is the good chimpanzee, let's say, it is the one that is not aggressive, we have a difference with the different ancestors that may be 8 million. There are some controversy in this data regarding, for example, the monkeys. We don't have the signalizers here, but the ones that we use a lot in the research lines, that is rhesus monkey, we have a distance from the common ancestors of 25 million years. It is translated in a noble part of history that is genetic homology. The closer to the ancestor, the more homology we have, more physiological characteristics, more characteristics making experiments closer to the ones carried out in humans.
Houve mais uma trava aqui. Ótimo. Agora foi. Dividimos os macacos em macacos.
We divided monkeys into monkeys from the old world, the Africans and Asian ones that are really the most substantial form used in experiments, and we have two representatives. We have the rhesus monkey and the cynomolgus, that is Macaca fascicularis, also known as the one who eats crabs. And those monkeys, in fact, from Asia and Africa, are the core of our scientific experiments. And we also have the monkeys from the New World that, for some interesting reasonability, they don't respond very well all the time because their phylogenetics is distant from us. We have about 35 million years from the common ancestor, so a chimpanzee 6 million years apart. If we think about a gorilla, 7, 8, 9 million years, but when we think about the rhesus, 25 million, and when we work with neotropical monkeys, 35 million years, or even before, in terms of comparing to the common ancestor. So it shows a genetic homology that is not the same. Although we have the genome that is similar, but we don't have 93% of homology. This is a noble sign for you to know which experiments may or not be similar to those carried out with the characteristics in human beings. This relation is changed today into a kind of a villain because thanks to the genome research, basically all animals from the new and the old world are now close to a definition, favoring scientists to find out which are the best biomodels to be used in the future. In our unit in Fiocruz, the ICTB unit, which is the Institute of Science and Technology and Biomodels, and they are responsible for the production and provision of biomodels. We have there, and I put here, if you work from there in ICTB, probably you will correct me because, again, the review was not good. We don't have Colitrix jactus, but they have Syrimis scienturus. I should have removed this one, but I would like you to understand the amount of non-human primates present in Fiocruz. In fact, they have specific areas to raise the animals, and obviously animals from the old world and the new world have their own peculiar characteristics in terms of being raised and handled so that they can be used, in fact, in many different researches.
Agora eu fiz uma coisa muito grave. Provavelmente, e não é que nós chegamos.
And here, the most important homage to the animals that are their attributions. All researchers must be remembered to that because of rhesus monkey and fascicularis. Probably you all followed the polio vaccination with the Sabin, which started in the 50s, with a vaccine that is attenuated against the 3 polioviruses. This number is being mentioned now by me, because some people contestate that, but I would say that from 1950 to now, about 1.5 million monkeys from the old world were used only for the polio vaccines. It's just for you to understand the reach of these animals in decreasing and curing diseases that in fact were a big problem to the human race. We have an obligation from the point of view of animal history, that is Rodrigo's obligation, and all of you working with animal research, to use all the forms and the ways to comply with animal ethics and welfare. So if you work with animal experiment, you need to know that the 3 R's are mandatory in the ethics policies to use any kind of animal, lab animal, since the small rodents of my friend Rodrigo and the big African primates. So we have important dating so that you replace and you reduce and you refine your experiments. All the experiments. Okay, now it's time for me to drink water.
Eu esqueci de tomar droga hoje. Isso é que dá ter plano de saúde.
With health insurance, we're immortal. That's important to clarify. Some surgeries, just 16, making us work and look alive. Continuing, there is no possibility that any of us be self-taught relating to experimentation with animals. All of the guides that are produced as norms should be carefully followed. So here we have the norms for the conduction of non-clinical or pre-clinical works, which are very up to date, and they encompass all the species that you could be working with right now. Something else that is essential inside our comprehension, especially economic comprehension, is for you to understand that any experiment that uses non-human primates, the involvement of a cost or price is based on whether the animals are SPF. So if we work with a rhesus SPF that is within these possibilities, you will spend about $8,000 or $9,000 for each rhesus. And that's a demand for the production of vaccines, drugs, neurobiological experimentation. You can only use animals that are SPF. And this cost of an SPF animal demands that the lab or the creation center be accredited. And this is another very serious issue. You need to have these credentials for the production and supply center. So with any kind of research that you conduct, for example, in biomanguinhos, 20 rhesus monkeys are necessary. You would already start with a cost of $160,000 just to have these SPF animals. And that's a bit of the reason of why we have 58 protocols in 38 years of work. The lab was constructed in 1982, and once again it was expanded in 1998. Today the lab only has 420 square meters. We can maintain it in a much simpler way. It's a very big area, but the demand is that this vivarium of creation should have negative pressure, control for access, operational procedures, just like any small or big lab for experimentation would have. And this demands that you should spend your day, your whole life really, just to get a lab like this to be in the adequate conditions for working. This is a simple presentation of a lab, experimentation lab with primates. This involves specialized colleagues, the correct procedures, specific cages, monkeys that have already been screened for these diseases that you are presenting at the moment. For example, if it's Zika, they have to be serologically negative for Zika. Chikungunya, they need to be serologically negative for chikungunya. And you need to consider this for other pathogens. The lab consists, as I said, it has been here since 1982. We've been working for 35 years with many units. As I said, as Rodrigo said, production labs, development labs that make us have a great amount of activity. And this is the location by Avenida Brasil, surrounded by small units that always pollute our air. Here we have the INCQS, and this is our unit. Leaving from Avenida Brasil and entering, we have quick access here, then to LANIL, which is the neurovirulence lab. The studies we conduct involve the viruses. In the studies, we can see if the viruses can create issues in the nervous system. These we need to identify if they are arboviral vaccines, yellow fever and dengue, mumps, rubella, and measles, or other viruses that could have a neurological implication. So effectiveness studies and immunogenicity studies involving hepatitis A, C, dengue, Zika, parvovirus. So in our job we use primates. We conduct the intracerebral inoculation with a neurotropical virus or a virus that in these animals provokes a clinical condition that can be compatible with one that is presented in human beings. The main protocols, international protocols conducted, we started in '82 with Japan, polio, then Canada. We don't have to read all of this, you know what's going on here, but just so you can see that we have had an international presence based on our researchers who are no longer with us. Some are already retired, others have left the institution, but they gave us the opportunity during these years to participate and to obtain knowledge about a number of diseases, like for example working with the yellow fever vaccine, and this was a great start for all of these protocols. We also had Brazilian partners that participated actively with the neurovirulence lab. In 1985 we started, and then we never stopped. Osvaldo Cruz, UFI, bacteriology, and other agencies, they all worked with biomodels, non-human primates. So all of these protocols that you see here are actually conducted by a large group, including my friend Marcos Freire. Is he here today? No? So he asks me for the following: don't forget to mention that all the protocols that are conducted by you at LANEU should mandatorily be multicentric. There are many labs that are qualified to collaborate with LANEU for this protocol to move on. We are not totally independent to obtain things simply by conducting trials with animals and obtain results that are convincing. We need these collaborations with this big network of people. Some of you are here, and you need to know that you have to consider time, schedules, a lot of work. I know some of this work is very demanding. My colleagues are listening to me. We had about 600 days of work with no breaks for some of these trials. So within these 58 protocols, some experiments even lasted for 2 years. And I'd like to finish up with a little mural, people that many of you probably remember, and that were part of this trajectory. Of course, a tribute to the greatest experimental pathologist in history, Osvaldo Cruz. And some of the main actors in our story are portrayed here. Some of them, unfortunately, are not among us anymore. Thank you very much for listening, and I'll see you next time.
Muito obrigada, Dr. Renato.
Thank you very much, Dr. Renato, Rodrigo, for your words. Now we will move on to our pitching session. Let's take a small break for them to load their presentations. So we're going to take a small break so the speakers can organize everything here. So we're just going to take a small break before the next session, the pitching session. examining board, Marcia Lisala, who is the advisor for our technological development. Unfortunately, sorry, excuse me. Sorry, that's my mistake. He is in Europe. I'm just gonna, it's good for breaking the ice. His flight was canceled, so he will only be able to arrive after the event. He was supposed to arrive on Tuesday, and he had a number of issues with his flight, so he's in Europe. And he can't be here today. So we have Masarasawa, Vice Director of Technological Development. Also in the examining board, Eduardo Soares from Biominas, CEO from Biominas, also in our examining board, and João Miguel, who is an advisor for the Vice Presidency of Production and Supplies and Innovation of Fiocruz, representing Marco Krieger, who also could not be here. He is in Brasilia at a meeting, so I apologize for my mistakes here. And now we are going to start the first presentation by Ronaldo Sembio. You will have 5 minutes to make your presentations. So this is fast. You know the drill. So we also thought João Peroni from BNDES would be here in the examining board, but unfortunately due to his schedule he could not show up, and he will not be here tomorrow, so we will only have 3 people at our examining board, those who I introduced to you. Thank you for being present here, and we will start with Ronaldo's presentation, SYMBIO. Good morning, everyone. Good morning, everyone. My name is Ronaldo. Thank you for the opportunity of presenting SYMBIO. I'd like to start making these questions for when we work with immunobiologicals. Were they produced in the correct manner? Is the sequence of amino acids correct? Is the three-dimensional structure correct? Are they contaminated with other macromolecules? What is the degree of purity of this immunobiological? Are post-traditional modifications correct? Because usually these molecules are produced in mammals and need to be glycosylated and other modifications. The different lots, are they reproducible? Are the biosimilar identical to the original product? So to answer these questions, you need to go through analytical methods that are very specific and precise. In this case, I coordinate this, and this technique is the only one that can answer all of these questions. It is a mass spectrometry technique, and it can answer the questions in a specific way. So within mass spectrometry, we can analyze proteins, lipids, sugars, organic compounds, analytic method that is very powerful, precise, sensitive, and that can answer the questions that I made in the first slide. At SYMBIO, we have 4 mass spectrometry equipments that can answer these questions, and we can conduct a series of analyses for identification of molecular mass, fragmentation, imaging, analysis of lipids, and quantification. And in this equipment, we have AutoFlex, IOTrap, and other pieces of equipment. More recently, we acquired the mass spectrometrist called Solaris, which is cutting edge in Latin America. It is used normally for this kind of analysis, and this piece of equipment is available at SYMBIO for the users. It can conduct a variety of chemical analysis, especially fragmentations, and determine the exact mass with great accuracy. We also have the QTOF, which can analyze intact proteins and other elements. We also have equipment, Maldi Tof that can be used, and the ion trap. All of these pieces of equipment are part of SYMBIO. The center is a facility that we are used to, just like those in the US. Users show the samples, they have the results and pay for them. So this is done in a very professional way, and it is available for the scientific community. We have a partnership with Biomanguinhos, with Fiocruz, and a variety of researchers already use our facilities. They are our partners, and I want to show you that this exists. It is at UFRJ, it is available, and we are open to starting new partnerships and making this technology available, which will be extremely necessary, not only for advances in characterization of biopharmaceuticals that we will start producing here in Brazil. Thank you.
Falei quase 4, por favor. Não, é porque é muito claro. Aqui, ó. A televisão é muito clara, ele não aparece na TV. Ó. Aparece, cadê? Só aqui. Aqui aparece, aqui esse também aparece aqui, ó. Aqui não vai. Agora tá indo mesmo. Agora tá indo? Já que tá. Bom, vamos lá. Muito obrigado pela apresentação, Ronaldo.
Thank you, Ronaldo.
As perguntas vão ser ao final da sessão.
Will be in the end of the session. Everybody will present.
Obrigado pela participação.
Thank you for your participation. This is the certification. Anyone who presents here will have their certificate. Now, Juliane, come to your presentation.
Para a empresa Novas Ferramentas.
Gianni, for the company, do you have a company?
A UENF,
okay,
UENF vai apresentar novas ferramentas
the novel immunochemical tools for differentiation of tuberculosis and non-tuberculosis microbacterial diagnosis.
Bom dia a todos.
Good morning, everybody. The title of my work is the development of immunochemical tools for the rapid diagnosis of non-tuberculosis microbacterial infections and tuberculosis, and we are working on the labs of UENF. Tuberculosis is one of the infectious diseases who kills the most in the world, especially in developed countries, and in parallel, mycobacteria is increasing worldwide, both in developed and underdeveloped countries. It's important to emphasize that mycobacteria from the AVO complex are more prevalent worldwide. It is important to have a differential diagnosis of these infections due to the clinical and radiological similarities, as the treatment for the infections is different. And currently the main diagnosis methods we have is bacilloscopy, X-ray, tuberculin skin test, and they present the disadvantage of not being able to differentiate mycobacteria. We have the culture that is the gold standard, but it takes time for the diagnosis due to the slow growth of mycobacteria, and PCR, which is rapid, but we have sophisticated and high complexity equipment, which is quite expensive in low-income countries. As an alternative for the diagnosis, we have the immunochemical tests, which allows the identification of unique antigens or that are expressed differently from the bacteria. The antigen as identified as MTP is only secreted in mycobacteria, and it's used in the immunochromatography test for the detection of the antigen in liquid and solid cultures. The advantage is that it's a simple method with high sensitivity, but the disadvantage is that it identifies only the mycogenes of tuberculosis and not the other ones So to identify new antigens, especially for the diagnosis of infections, our research groups developed and identified the antigen APA mode from Mycobacterium avium, and we produced monoclonal antibodies against this antigen. And we could check that the monoclonal antibodies produced only recognize Mycobacterium avium and not the Mycobacterium tuberculosis when compared to the polyclonal antibodies, which recognize both complexes. In 2011, we published a paper on this data, and this way we produced a kit, a sandwich ELISA kit, anti-APA, that is for the identification of APA antigens in feces of animals with paratuberculosis and in different cultures of MEAV, and we published the patent, and we recently published a paper on the use of this antigen as a biomarker, and we applied the sandwich ELISA for the identification of the APA protein in the supernatant of cultures, and we could observe that the kit only detects APA protein in the supernatant of the culture of the N avium complex for tuberculosis and Mycobacterium avius anisus, and it doesn't identify APA for the Mycobacterium casaceum and Mycobacteria from the tuberculosis complex. So this ELISA identifies only APA in the MEAVs complex. So the next step is to develop an immunochromatographic test to detect the antigen in culture as it was produced for MPT64. The production of a multiplex kit based on the detection of antibodies for anti-APA and MPT64 that we are producing this monoclonal antibody in the lab, and assess the use of this antigen in the diagnosis of Mycobacteria infection ex pulmonary, as it was proven recently that MPT64 of Mycobacterium tuberculosis may be detected in these infections. And currently we have a sandwich ELISA kit to detect, and we tested in different clinical isolated, and our interest is to look for companies to help us in the development of the kit. We have a patent currently that is filed, and this is my contact email. Thank you for the opportunity.
Assim, Juliane, seu certificado. Obrigado pela apresentação, participação. Bom, oi? Foto? Pode ser. Cadê o... Tá. Bom, a próxima apresentação.
Next presentation is Thalita, from Thalita Souza.
Thalita Souza, da empresa aqui.
Do you have a company?
From
UNB.
Vai falar sobre fator de
and she will talk about the growth factor.
Tudo certinho, apresentação. Sim, só laser point. Laser point é só para explicar. Eu não vou precisar ver, só direito. Agradeço a presença.
Thank you for your presence. Have a nice presentation.
Bom dia.
Well, good morning. My name is Thalita,
and
this project of production of PDGF from platelets using mammalian cells is a collaboration with the Nucleus of Cellular and Molecular Therapy of USP, the Medicine School of USP. I work with them in the lab and development of some work in this project at the University of Brasilia. The first point to be addressed here is to know what is PDGF and what would be the main actions of this glycoprotein in some systems. First, PDGF is part of a cascade of blood coagulation and cicatrization, so it may be used basically for repair processes in the tissue and regeneration. Within this area, it can be part of regeneration of tissue or periodontal regeneration, cell differentiation, ocular neovasculation, coming from the principle that it is a chemiotatic substance, mutagenic and angiogenic, and it also has this angiogenic action. And what does diabetes has to do with the PDGF? Diabetes mellitus is a metabolic syndrome characterized by many diseases associated, and one of them is the peripheral neuropathy, affecting 50% of patients with diabetes. It's important to mention that 90% of the Brazilian population has diabetes, both type 1 or 2, and we believe that about 20 million people will be affected by diabetes in 2045. Currently, we have 851 million patients in the world with diabetes. Regarding the federal government, they spent $25 billion in the treatment of diabetes. To develop or to study better the production of this growth factor, we created this approach, and we used human kidney cells with antivirus carrying the construction of PLPDGF. And you have to remember that these cells are adherent cells, so we first had the process of building our strain and then establishing the strains in adherent bottles, then followed by the increase of scale for systems adhered to microcarriers and the characterization of our glycoprotein by SDS. It was followed by Western blotting, and then the quantification with the ELISA method with a commercial kit. Followed by some in vitro and in vivo assays that I'll show later. So this is our first construction, the first strain we created. Then we followed with a cell culture in bottle, and followed by spinners and wave. And you have to remember that currently we produce cultures in 1 liter of culture medium. Then we went through the purification stage. By affinity, we used Actor Purifier, and here we have the SDS page proving the presence of our protein and also Western blotting. And also here we have the biological activity of our extract, proving by timidina tritiated assays the presence of this activity. Some preliminary in vivo assays evaluating the cicatrization of wounds with PDGF, and these are the main goals, the main steps from now on. Thank you.
Obrigado pela participação. Próximo candidato é Patrícia.
Next candidate is Patricia. Patricia Prank.
Qual a empresa?
What is your company?
Da Federal do Rio Grande do Sul.
Okay, from the Federal University of Rio Grande do Sul. I'll take 1 minute and a half from Ronaldo. Anyway, good morning, everybody.
Ok. Bom dia, meu nome é Patrícia Prank, eu sou professora de hematologia da Faculdade de Farmácia da URGS. Estou aqui para apresentar projeto que, na realidade, eu chamei aqui como subprojeto, o Recupera-Med, mas ele já faz parte do projeto que eu chamo de Projeto Andar, que nós já trabalhamos no laboratório há cerca de 10 anos nesse projeto. Eu fiz doutorado nos Estados Unidos, há cerca de 15 anos eu trabalho com células tronco. Mais recentemente, há cerca de 10 anos, nós introduzimos a nanotecnologia e começamos a estudar mais a lesão de medula espinhal, que é
the lesions of the spinal cord with this project on there, and then we had the sub-project that is Recupera-Med. We also have the Institute of Research in Stem Cells, and our initial focus was to use stem cells for the hacromedular cells. We have great access to the website and patients, and we have lots of access every month. And in the Institute we have Drauzio Varela, Luiz Fernando Veríssimo, Cristina Ranzolin Falcão, many researchers and scientists and business people supporting the project. And I would like to remember that Dr. Maiana Zatz from USCIS were one of the authors, along with me, to approve the law for the stem cell research in Brazil. We have this law, it was approved in 2005 to use embryonary stem cells. And why am I saying embryonary? Because I would like to have embryonary stem cells to help patients. But then we started dealing with pharmacy and medication, and to my surprise, although we have more than 10 papers publishing showing stem cells as a good use and the best approach for medullary lesion, we had some other medications that was much better, and it was a big advantage because it's much easier to use a medication than a stem cell, and this is what I would like to show. Nanotechnology was introduced as the nano-delivery because for 10 years we've worked on that in the lab. Needless to mention that nanotechnology over the years is increasing a lot in terms of publication and prices and amounts that the fomenting agencies both in Brazil and abroad are dealing with that.
The spinal cord injuries, they confine people, a lot of times young people, they are confined to a wheelchair. In the world, we have around 2.5 million people affected by this, about 130,000 new cases per year. In Brazil, this is very alarming, 40 new cases per year per million of inhabitants, 6,000 to 8,000 new cases in Brazil of people who are confined to a wheelchair. And we know there is no cure for this. There are few medications available, few treatments to improve their quality of life. They say that any improvements for them help them, even just controlling their sphincter, anything like that would be great for them. Some results with mice have already showed improvement, which would be about equivalent to this. The patient can control their sphincter when before they were not able to. So we're trying to get these improvements, get these resources to accelerate these results. Laíse, this is Laíse, I met her. She supports our work because I'm starting a partnership with Miami University. She was treated there. She's a triathlete. She started this treatment with stem cells at the University of Miami a few years ago. So I can't reveal the name of the drugs, but the good news here is that these are drugs that are already used today. We are just presenting a new pharmaceutical formula. And what do these drugs show? What I'm going to show here in this graph is that 0 means no movement in the mice, and 21 means the maximum amount for normal movement. We always work with this control system, so if the mouse suffered this injury or partial injury, one that has spinal cord injury, and our treatment, there's always a spontaneous improvement in all of the mice. For example, this group of mice went from 11 to 16, so it's not perfect, but it's a significant improvement. In the second treatment with other kinds of drugs, this was a lot more visible. We worked with the brute form of the drug. Now we can see we're showing its efficiency. We're working with derivatives, purified forms. And we have partnerships with a lot of countries around the world. Miami University will be our partner. If we think about clinical trials, maybe we can do them here in Brazil, but there they have everything ready to conduct these kinds of studies. So the objectives are to continue studying, developing medication inside the hospitals, to do this business to business in the pharmaceutical industry. If anyone in the industry wants to support us, that's fine, and to improve the results. So maybe not obtain a complete cure, but improve their quality of life. That would be great. So to do this, we are doing molecular modeling. Oh, I don't have any more time. And the advantages of our products, sorry, I didn't have time to finish, but thank you. This is our team.
E agora...
And now, Ana Carolina from the SELP software from Unicamp to present the SELP software.
Nenhum desses do meio, que lá do meio vai travar a apresentação. Então, voltar, só se por acaso passar mais esse aqui, se precisar voltar, você volta.
Tá bom, então aqui.
Não tá no meio.
Só aqui, né? Tá.
Não tá indo, né?
Primeiramente, bom dia a todos.
Good morning to everyone. My name is Ana Carolina. I am a Unicamp student, and I'm here to present the SELP software counter of erythrocytes, leukocytes, and platelets. So around the world, we have around 7 billion inhabitants. 50% of this population lives in poverty, and 25.6% live in extreme poverty. So we are speaking about people who live with less than $1 per day. So let me ask you, if these people don't have money to eat, how can they take care of their health? On the other side, we have a different outlook. People who live in countries that provide some kind of health, total or partial, for the population of the country. Here in Brazil, we have the unified health system. 70% of the population depends on the unified health system in Brazil. When we speak about health, it is important to highlight the medical diagnosis. 70% of medical decisions are taken based on lab exams. The hemogram is the most demanded one. Currently, the hemogram is made with 3 methodologies, the manual method and the automatic method. The manual method has a low cost, low speed, and low accuracy. Automatic method has high cost, high speed, and high accuracy. So you must be asking yourselves, how high is this cost? Before I started this project a while ago, I called a lot of representatives of hematological equipment, and I saw that this price is about 25,000 to 90,000 reals. So why can't we create a third methodology with low cost, high speed, and high accuracy? And why can't we do all of this through a simple computer? The answer is yes, we can do this through the SELP software. When it is concluded, it will be a computer program based on digital image processing techniques, where its innovation is counting simultaneously the 3 kinds of blood cells in the pathological and non-pathological state in the Neubauer chamber. And here I have some images of how this algorithm works. Our potential market in Brazil, we have about 200 million inhabitants, where 70% of this population totally depends on the unified health system, the SUS. So it's an essential system. Any methodology that can reduce costs is always very welcome. in the context of private hospitals, they are 1/3 of hospitals in this country, so we can use this methodology as a method to confirm this. And since we are speaking of reducing the cost of methodology, automatically the price of my service will be lower. It's more accessible to less favored populations, indigenous populations. And in this idea of reducing the cost of the methodology, we are indirectly influencing entrepreneurship, especially young people who are finishing college now, want to be entrepreneurs, but they think it's expensive to pay for equipment. That's over 25,000 reals. Our business plan would be to start sales by e-commerce. We are thinking of about 5,000 reals, which is a lot cheaper than conventional equipment. And in this first year, we would like to reach 3% of the market. This is my team. I have a biomedicine degree. I currently I'm doing a graduate program in electrical engineering, and these are the people from my team. They are all masters or PhD, and they are professors at state universities. Our objectives for 2019 are to develop the algorithms that will count these cells in pathologic and non-pathologic states, test them in terms of accuracy, specificity, and sensitivity, and in different kinds of computers. Our objective here at the Innovation Hub is to look for intellectual and financial partnerships that can help us to validate this methodology when it's finished, and that can help us to put this product in the market, because that is how we can get our objective of helping people with low income to have high quality health. Thank you so much for your attention.
Só pegar seu certificado. Passa seu certificado. Obrigado pela presença.
So now that the presentations have finished, we are opening this up to questions. If anyone here has questions or comments, feel free to make them. Any questions, comments, issues? First of all, of course, the examination board is the examining board is the priority. Any questions? Any considerations? Well, congratulations to everyone. First, you need a lot of courage to do these pitches. You're exposing yourself. You need to know what you want, what your objectives are. So congratulations to all of you who were willing to do this. Thank you for your time. It's so important to be here and look at new ideas, because I think our objective, we all have the same objective. We want to work for the population's health. First of all, I'm not going to make comments on the presentations. I don't know if this is the moment, but I'm going to make a very specific question to Patricia. These drugs that you cannot reveal, but that they are already in the market, are they synthetic or biological? What are their bases? Well, they are drugs that we use in other diseases. The innovation is using them for spinal cord injury and the new formulation we are creating. And will it be necessary to conduct clinical trials for this new therapeutic use? Yes, I think so, because it's different information, but it's already very well accepted for a disease, and I think the clinical trial will not be difficult. Andrea, are we going to have a moment for comments later? Okay. So generally speaking, the presentations were good. I thought in most presentations there was a lack of a business plan, a structured business plan was missing. Maybe that's just my perception because it's my area. I look at the business part of things, but I thought that was lacking. But in general terms, I was able to understand the proposals, the objectives, and what you're looking for here. And this allowed us to be able to analyze the viability, how this will be developed, if it's worth investing, and if it's possible to work with a partnership to be able to launch these projects. So congratulations. I hope everything works out tomorrow as well. Well, first of all, let me stand up. I want to thank everyone who made a presentation. For those of you who know me a bit, I make my life doing this. I listen to people's pitches. So I'm kind of trained to understand and listen and see what you want to say, what you should say, and what you shouldn't say in a pitching session. In general terms, I thought there was a lack of a business model. I think you should think of a business model. And there are many ways to do this. Some people have been through our program, so they know how to do this, right, Ana Paula? You know about this very well. Something that's very important and that most projects presented forgot about it, let's say, and it's essential for us in the examining board and for potential investors who are here in our audience, is the team. Most of you did not speak of your team, and the team is essential here. You need to speak about who you're working with, who's part of your team. So if any of you will present tomorrow, please add this section about your team. And review the market issue. Who is your market? Who are you selling this to? You use too many technical slides, so you should reduce the number of technical slides. Just use the direct information, the basic concept, make it fast and focus on the market, how you're selling this, how you expect to sell this. And you might not even have the correct model, but it's good to at least have a model to present, and then maybe somebody will review this and give you some suggestions about what can be done to improve the project. But in general, these are my comments, but the projects are great. I enjoyed it very much. Well, in name of the Vice Directory of Technological Development, I'd like to thank everyone's participation, the speakers as well, and I missed this. I thought you were lacking the same issues that João and Eduardo mentioned. We know how it's difficult to work on development here in Brazil. It's a constant struggle. So I'd really like to congratulate all of you for this initiative, and we are here. We will be here tomorrow as well. So any other observations, comments? Thank you for your presence. We'll see you tomorrow for the next pitching session. Thank you for your presence.
Obrigado.
You see here that we have three kinds
instrumentos,
of
grosseiramente.
instruments.
Nós temos os grants
So
para
we
ICTs,
have the
os não
grants for
reembolsáveis para
ICTs,
os pesquisadores, os
the
não
ones that are
reembolsáveis
not
para as
refundable for
empresas, a
researchers,
subvenção
the one
econômica,
refundable for
e
companies,
os
and
empréstimos
the
subsidiados.
low ones that
Você vai
have
ver que,
subsidy.
ao contrário
As
do
you
resto do
can
mundo,
see,
nós
unlike the
utilizamos
rest
muito
of
mais
the world,
empréstimos
we use
do
much
que
more low ones
os recursos para
than
ICTs
the
e subvenções
resources
econômicas
for
para as empresas
ICTs or
para
grants
desenvolver
for
inovação no Brasil.
companies to develop
Isso fazia algum
innovation in
sentido
Brazil. It made some sense
quando nós tínhamos taxas de juros de
when
20
we
e tantos, 30 e
had
tantos por cento ao ano e a
rates
FINEP emprestava a 3,5%, 4,5%
of 30% a year, and we load at
implícito gigantesco.
3% a year.
Então, essa não é mais a realidade do país. E acho que
This is
esse é o
not the
grande desafio que
reality
se
of the
impõe.
country
Como
anymore
é
in
que
terms
num
of
momento de
interest,
restrição de
because
recursos
we
que nós
have
estamos vivendo, como é que a gente
a
faz
restriction
para
of
que a
resources
inovação não
here,
pare?
so we
Como
would
é que a
like
gente
to
faz
know
com que os
how
senhores
to
que são
work on
mola
innovation
propulsora
without
daquilo
stop
que se
and know
desenvolve nesse
how
país,
you
continue
who are the engine
suas
for
inovações,
that to
desenvolvendo
keep
seus
on
projetos
developing your innovations and
consigam
projects, and
colocar os produtos
that
no
you
mercado.
can bring products to
Então,
the market.
FINEP é uma empresa pública que atua em
FINEP
todos
is
os
a public
segmentos
company
da
working in all the
processo de
areas
inovação,
of the innovation process.
Desde o TRL
Since
até o 9, então a gente financia de pesquisa
TRL1
básica,
till 9,
até
so we
o lote
finance
pioneiro
from
colocado no
basic
mercado.
research until
A gente
the
tem
pioneer
instrumentos para
batch
cada
of the market,
dessas
and
etapas.
we have instruments for each one of the
Então,
stages.
nós temos ali
So we
na
have,
ordem, né,
in order,
a subvenção para as ICTs.
the grant for SATs.
A gente chama muito de ICT cooperativo. Lembra que o
And
vídeo
I'm
falava
sorry,
de 700
they are
projetos em cooperação,
cooperatives,
tal.
and the video
Então,
mentioned the
instrumento
700
que
projects
usa
in
para estimular a
cooperation. So this
cooperação
is the
no mercado
instrument
privado.
to stimulate
A
cooperation in the
subvenção
private
econômica,
market.
que são os editais
Economical
de
subvention,
subvenção para as
these
empresas.
are the call for bids
O
for the
instrumento
companies,
cooperativo,
the
o
cooperative
investimento em
instrument,
fundos e equity, vou falar
investments in
pouquinho aqui
funds
do
and
FINEP Startup,
equity.
que é o
I'll talk
instrumento
about
que está mais
FINEP Startup, that
latente
is
no
the
momento,
one
os
that is more
empréstimos
latent in the moment.
subsidiados, a gente faz isso
And
de
the
forma direta ou de forma
subsidized
indireta.
low
Então, a
one.
gente tem agentes
And we
regionais,
do
nós
it
temos bancos de
directly or
desenvolvimento regional
indirect,
que
so we
operam em
have
linhas da FINEP
regional
com taxas
banks
até mais baixas do
operating
que
with
operam diretamente.
FINEP with lower rates than they
Então,
operate
como é
in
que a FINEP
general.
avalia
So how do we evaluate
uma proposta?
the
Acho que isso é uma
proposal?
coisa
I
interessante para
think it
quem
is something
quer se
interesting
conectar
if you
com
want
um
to
agente
connect to a
público. Nós
public
usamos
agent.
dois
We use
grandes
two main
parâmetros. No primeiro grau de inovação,
parameters.
o de laranjinha ali,
First, the
e o
degree of
segundo,
innovation
relevância.
in orange, and the
Então,
second one, relevance.
eu deixei até, depois eu peço que disponibilize a apresentação.
I would
porque
like to
você
make
fica
this
anotando,
presentation
fica
available
preocupado
for
com
you
tirar
later
foto, fica
on,
tranquilo,
so
que
don't
fica
worry
pra
about
vocês.
taking pictures
A gente tem
or
aqui, eu botei aqui
taking
uns
notes,
key
don't
indicators
worry.
aqui, que é para
I have the
facilitar
key indicators here
quando
just
você
to
quiser
make it
conversar sobre uma
easier
proposta, porque a
if
gente
you
olha,
want to talk about
mas
a
a gente olha,
proposal to understand what we evaluate.
muito a
We
questão do esforço,
analyze the
do risco tecnológico
effort,
envolvido
the
naquela
technological
proposta, o
risk
esforço a ser
involved
feito
in
para
the
a consecução daquele
proposal,
objetivo.
the effort
E a gente olha, isso
to
é
reach
importante
the
para quem?
goal. And this is
Relevância, é
important,
muito fácil
this
resumir
is
relevância. É
relevant
importante para quem? É importante só para você?
only for
É
you,
importante para a sua
or
cidade?
is it
É importante para o
important
país? É
for
importante
society, for the
Isso
country,
muda a
for the
maneira como qual
world?
conselho
It
de
changes
recurso,
the
o quanto
way
conselho de
you
recurso.
get the
É assim que a gente
resource or
avalia
how much you
na
get.
FINEP.
This is our evaluation at FINEP.
Lógico que tem toda
Obviously,
a questão de
we have
correlação com política
all
pública,
the
isso também
matter
pesa, né?
of
Então, às
public
vezes, uma coisa nem é tão
policies.
inovadora,
Sometimes something is not
mas
that
você
innovative,
tem
but
alguma
you
conexão com
have
política pública,
some
a FINEP acaba
connection
entrando
with
para
public
fazer só o
policies,
escalonamento
and
daquela
FINEP
inovação,
comes here just to
daquilo,
scale the
ele até já está
innovation,
começando a entrar no
because
mercado, a gente
probably
apoia
it
para poder
is
dar uma
entering the
difusão
market,
tecnológica.
and we
Enfim.
support to give the technological
Então,
spread.
falando primeiro aqui dos empréstimos
So, talking about the low ones
subsidiados, vou falar bem
that are
rapidinho. A gente, como eu falei, a gente opera via agentes
subsided.
centralizados.
We have centralized
A
agents.
maioria dos senhores são de pesquisadores.
Most of you are researchers or from small companies, so we work with BDMG, Age Rio. You have some logos there to see, and we operate indirectly with the companies with gross revenue of less than 90 million, proposal less than 10 million, the interest rate TGLP or TJLP plus 1, it depends on the proposal, because more than 16 million, it's up to 90, and we have the grace period of 24 months, and the total term is 96 months. We have now a new line being entering on the market.
conectado a uma proposta
that we are
de
connected
inovação, a gente
to
vai
an
conceder
innovation
30%
proposal, and
a
we'll
30%
have
do valor financiado
30% of the
para ajudar no
value
giro da
funded
empresa.
to
Porque
help
muitas vezes a
the
inovação
company,
não ganha
because many
escala, não é porque o
times
empresário não sabe ser
innovation
empresário.
is not
É
scaled
porque está
because
asfixiado com uma série de outras questões. E a
the
literatura mostra
company is
que
dealing with many
a
subjects, and
sobra
in
de caixa do dia
literature
a
we
dia
see
dele
that
é fundamental para ele continuar
the extra
inovando.
money they
Então, a
have is
ideia
important
por
for
trás do instrumento é
the
essa.
company
E a gente
to
tem
keep on
condições especiais
innovating,
para
and
quem
we have
investe
special conditions
15%
for
do
those
seu,
who invest
do seu orçamento
15% of their budget
ICTs. Chama
in
FINEP Connect. Eu vou falar um pouquinho mais para frente.
ICTs, and I'll talk about it later on. It's
Aqui o empréstimo direto, aí são empresas
Here is the
acima de
direct
90
loan
milhões,
for
com taxa
companies
de
over 90
mais
million, with the
3,5,
rate of
condições
0.5 to
diferenciadas para quem trabalha com
3.5%,
telecomunicações, tá?
differentiated conditions for different
recebem
companies, like
remuneração
if you
fixada em
work
TR,
with the Fontel,
então,
you
é
have in
TR
a
mais-
fixed way, so you
TR mais
have
3, TR mais
TR
5, então muda
plus
bastante a
3 or
condição.
plus 5, it changes the
E com
condition.
uma carência de até 36 meses e
And
também
with a
condições
grace
especiais
period
para
of
quem
36
conecta
months and special conditions for those
com ICTs, 15%
who are
conectado com
connected
ICTs.
with ICT
Por que isso é
and
interessante? Por exemplo,
50%
vocês viram no
connected to
videozinho
it.
ali, uma
It's
pessoa com
interesting
uniforme da prática, uma
because
luz,
you saw
ela tá
someone in the
cooperando
video
com
with
a
a
USP, que são
uniform
jornal,
cooperating
muitas
to other
vezes,
person.
na utilização das
And
moléculas
it's
do
common
canabidiol
to
para
see,
sistema nervoso
like
central.
for example, the
Então, ele
study
acessou,
of the cannabidiol
viu como ele acessar
molecules.
essa
So
linha, né,
you
investir
can
uma quantidade
access
de
this
dinheiro
line
bastante
and
relevante,
invest
bastante
money that is
relevante
relevant. This is a
para as
work we do
universidades.
for, and we help the
Então, para as
universities
pesquisas, o que a FINEP está
in
dizendo,
terms
com
of
toda
research.
clareza,
So
traduzindo
what
aqui,
we are saying
é
in
que
translation
essa
here
esse
is that
relacionamento ele é
this
desejável, ele é
relation is
incentivado.
desired
Isso traz
and
valor para a operação perante a FINEP. Isso aqui dá
leveraged,
condições
so
diferentes.
it brings interest to
Então,
FINEP. So we have
a
different
cooperação, ela
connections.
tem que ser
So the cooperation should be at least 15% of the amount of the project. If it's 15%, I will finance up to 100%, and I will take a percentage of this basic rate, and I will do this as it goes up. So 25%, then I increase this grace period to 60 months, and so on. And so you have these different deadlines. I'm just going to go quickly through here to say that what FINEP finances is more innovative than what other companies are doing. So in the national market and the global market, what has resources from FINEP compared to FinTech, which is the research, FINEP has a lot more reach. So this is the moment of FINEP. This is our startup, and we want to transform what is research, prototypes in a pre-commercial phase. So you want to support someone who has a competitive prototype. You have a prototype that can be presented. You can connect to that prototype and connect it to another business to make this scale doable. And that's the idea, scaling the project. So just to give you an idea of the instruments, we analyze the investment proposal. We have the evaluation board, and lots of companies participate in this evaluation. We have a technical visits and legal analysis, $300,000 to validate the prototypes and make them commercially viable. So the target of the product is to work with those who have $1 million of revenue, and this is limited to up to 6 months before you can't create it after the open call, because then it gets messy. So just to give you a general idea, this is the message that I want to get to you. The selection process, there are 4 rounds of evaluation, 1,686 proposals submitted, capital demand of 1.6 billion reals, and we launched them through investors and other agencies, 7 million of private investment, 10 companies that received support from FINEP startups were supported by FINEP in another moment. So we're not simply abandoning this business. Here's your money, get out of my way, okay? That's not the idea. And we are involving 143 specialists who participated voluntarily. Nobody won 1 cent for participating. Nobody was paid. This is a market that is maturing, the startup market, and people are volunteering to participate in it. And within the startup area, we also collaborate with other agents in the sector. So I'm running out of time. I just want to give you a brief idea. So this is a 4 over here. In the current FINEP portfolio, I have 4 biotech companies and 6 health tech companies. So I have similarities here. I have biotech and health tech, but they're separated here because of our internal workflow. 49% are in tech diligence, 7% are contracted. So in tech diligence, we have to see if it can be supported. And then we have the legal analysis to look at the whole process. So we participate in the results. So if you're going to receive $1 million that are non-refundable, that becomes refundable if you can commercialize this. So FINEP has the mission of playing this role. I was speaking to Eduardo about this. This is a big role that we will try to play. So what we can finance, it's a little bit of everything that has to do with innovation, with FINEP Connect or any other instrument in FINEP. We can finance anything that is related to the proposal of innovation. Of course, the non-refundable resources have some restrictions that have to do with legislation, but our idea is to be able to finance anything to help innovation. So that's our mission. That's what I wanted to say. 15 minutes and 44 seconds. Thank you. I told you I was disciplined. Now we would like to call the representative of Advent Capital, Director of Advent Capital. Ótimo. Obrigado a todos.
My knowledge of Portuguese is limited
Eu não
to
sei.
obrigado, so I will be speaking in English today. So my name is Kabir Aziz. I'm coming from the Global Health Investment Fund, which has now been rebranded as Advent Capital. So apologies in advance if I interchange the 2 names throughout the presentation. So what is Adjuvant? So we are a life sciences fund that was established by the Gates Foundation in 2012 with the idea of using traditional financing mechanisms to fund the development of drugs, vaccines, and diagnostics focused exclusively on conditions that burden low and middle income countries. And the real impetus for this was that historically these types of products have been almost 100% grant funded or funded by non-dilutive funding. The thesis was that there are a subset of these products that actually have compelling underlying business models that should be able to generate compelling financial returns for investors. So in that sense, if we can show that there's compelling business models here, we can crowd in private capital beyond just grant funding and expand the capital base that's actually going into global health. So Adjuvant is designed as a classic double bottom line private equity fund with 2 very discrete objectives. One is to earn a financial return for our investors, and second is to show measurable improvement in public health in low and middle income countries. Now, prevailing wisdom would suggest that these 2 are opposing viewpoints. Our view is the opposite. We believe that if you are identifying the right opportunities, if you are structuring investments in creative ways, these 2 perspectives can actually work hand in hand and enhance each other. So what does that mean in terms of what we invest in? We have a very specific mandate that's been set out by the Gates Foundation and our other stakeholders. And that mandate is to focus exclusively on upstream innovation. And what that means is that our financing goes towards clinical development of drugs, vaccines, and diagnostics, focused primarily on infectious disease and maternal and child care. In terms of our product development risk tolerance, we are not traditional venture players. We operate more like a growth equity fund, which means we typically look at assets in phase 2 development or later. What that means is that the products we look at must have a compelling safety profile, as well as some signal of human efficacy before we invest. Ideally, we want grant funding to take care of stages 1, 2, and 3, and then venture funding such as us to come in later stage and be the last money in before the product comes to market. And this is just, will give you a flavor of our current portfolio and the projects we've invested in. For instance, we are super proud of the cholera vaccine that we supported back in 2014. This cholera vaccine was developed by a South Korean biologics company and is now the most widely used cholera vaccine used in outbreak and endemic settings today. In fact, it was just recently distributed to Mozambique post-cyclone, where they're seeing very high incidence of cholera over the last few weeks. Similarly, we're super excited about the chikungunya vaccine we supported a couple years back. As you guys might know, there is no vaccine and no direct treatment for chikungunya, and we believe this could be the first vaccine to come to market. And as you can see, just generally from the portfolio, we are very much product focused. We will custom tailor our investment instruments, whether that be preferred equity, convertible debt, or project finance, to meet the needs of the product. We want to find ways to bring these products to market as quick as possible and are agnostic as to what instruments we use, as long as we're maximizing return and mitigating risk and doing what's best for the counterparty. So shifting gears a little bit, I wanted to talk a little bit about this idea of building trust by investment and building partnerships and how we think about this and how we see it going forward. And I'd like to point you to a specific case study that we've encountered over the last couple of years. A couple years back, we were spending a lot of time diligencing a very promising vaccine startup focused on infectious disease. And we spent months diligencing it. Ultimately, we led their Series C preferred equity round and funded their lead indication. What's interesting is that simultaneously, there was a public health funder also doing diligence on the company, and they ultimately ended up funding that company's early stage pipeline, focused on earlier stage infectious diseases. So ultimately, this was a good outcome for the company, and it was a good outcome for public health, because a number of essential vaccines received the funding needed to advance in their clinical development timeline. We internally thought, this is great, but is there a better way to do this going forward? Is there a way to collaborate and promote coordination among these different organizations so that we can more efficiently get capital into essential public health programs? And just to give you an example, we and this other public health organization hired the same exact technical advisors to do scientific diligence. So what that means is that you had redundant transaction costs, you were burdening the company with increased diligence requests because you had two organizations sending in the same requests, basically. And I know that sounds maybe trivial on the surface, and maybe that doesn't matter, but what we have to realize is that there are only so many dollars going into global health R&D for the developing world each year. Each year we hear about the fact that there is a $1 billion plus shortfall going into this space. And now that's not a problem that we're going to be able to solve today. It's not a problem that we and one other public health funder can solve on our own. But what it does mean is that we need to take the dollars we have today and stretch them as far as we can. And to do that, we need to encourage more collaboration among funders. So we are in the process now of actually formalizing memoranda of understandings with a number of public health funders. We executed our first one earlier this year and are working on a few more this year. And the whole idea behind this is that we want to promote the sharing of information. So there's really three key facets we see for providing this basis for cooperation. One is to regularly share information on our portfolios and our pipelines to various funders. The second is to jointly network within the public health funding space so that companies are aware that we're working together and that we can maybe jointly diligence the landscapes that we're interested in. And finally, the ultimate goal is that, can we identify the most promising technologies in global health and fund these projects together rather than working in these really separate, discrete work streams. And ultimately, we think that these partnerships are a way of generating synergies in the global health funding space and act as what we call a force multiplier for advancing global health. So our challenge over the next few months and coming years is to, one, strike up these formal partnerships with public health funders, two, to execute on the objectives that are set out by these MOUs, and finally, as I said, ultimately identify the most promising technologies for investment. And we would encourage all the other funders in this room and this space to take on that same challenge, because we truly believe that radical transparency is the future, and that with radical transparency among funding groups, not only will that encourage better and faster innovation, but it will also just be a more efficient way to allocate capital across the market. So I'll wrap it up there. But before doing so, I will shamelessly plug our organization and say that we are always on the lookout for new opportunities, ideas, collaborations. So please don't hesitate to reach out or find me after the session. Thank you.
Na sequência, vamos chamar o Dr. Jorge Guimarães, diretor-presidente da Embrapi.
Boa tarde. Boa tarde. Muito obrigado pelo convite. Acho que o Rodrigo já deu o caminho das pedras. Esses eventos não têm empresa.
We don't have companies in the PD business and therefore capable of promoting competence that our institutions have and that Fiocruz is showing and Guimarães is showing so clearly. One event like this, as we see downstairs in the main room, if it were a developed country, more than 1/3 would have people here from companies, and this is our case. That's why Embrapi was created, for the innovation in the company, and it differs a lot from the other models we have. I think we have a lot of slides here for a short time, so we'll be brief here. We have all conditions to have innovation in Brazil. High scientific production, however, with low presence in the technological area, lack of R&D centers in the companies. I mean, we lack that. It's not that it's deficient. We lack. We have 200,000 industrial companies in Brazil. This is self-declaratory. We don't know if it's true or not. But anyway, 1,200 for 200,000. So we have more than 90% without R&D center. So we don't have qualified people. We have no applied research, and therefore no innovation. We don't produce patents. Do you know the Brazilian patents? Most of them are in the universities and the research institutes and of the companies. So Embrapi wants to break this pattern. We have a lack of technological development, so we need to create a new model and story, because we cannot lend money to companies for innovation if there is no PhD center, so it won't work. So this is something that we are facing yet. There is this perspective that I know since I was a little boy of So, do you know the
25
effort to go
bilhões de
from
dólar
1.2
novo por ano.
to 2? It's $25 billion a year. Each 0.1 is $3.1 billion.
E não será com dinheiro público.
And it won't be
obviamente.
with public money.
Os países que estão lá,
Obviously, the countries
acima
there
disso, é 70% pelo menos de empresas.
have at least 70% of
são
companies.
nossos
So
desafios.
these are our challenges.
Além disso,
Other than that,
junto com isso,
and
7
along
décadas,
with
mais
that,
de
we
7
have more than
formando
7
recursos
decades
humanos de alto nível.
training
A gente tem
high-level
produtos
human
de muito boa
resources. We
qualidade.
really bring
E
quality and good people to the
capacitando, nós
market.
temos
And
na Embrapi
in terms of
visto isso.
training, Embrapi
Quantos
has seen
grupos
this,
estão
how many
altamente
groups are highly
capacitados
trained
recurso humano,
in
equipamento,
human
infraestrutura,
resources and
etc.,
equipment,
para fazer
infrastructure,
trabalho
and
com
so on to work with companies.
Bom, a Embrapi não é órgão do
Embrapi is not a governmental
somos uma
body.
organização
We are
social
a social
muito
organization that is quite small,
pessoas,
only
privada,
20-something people.
que
We are a
presta
private
serviço
organization
para 3 ministérios:
working for 3
ciência
ministries:
e tecnologia,
Science and
educação e
Technology,
saúde.
Education, and Health. Health is the last
Nós
one to join the
selecionamos
group.
unidades,
We selected
chamadas
units
Embrapi, são
called
grupos
the
de
Embrapi
pesquisa
units.
de centro de
They
pesquisa
are
de
research
universidades
groups in research centers,
têm
universities,
vocação
with the vocation
teogeniza de trabalhar com empresas.
to work, and they hate
experiência.
companies, and they have
nós
some
não somos
expertise because
CNPq,
we are not CNPq and
FINEP,
FINEP and
Alfapeg,
CAP
né?
and Alfapeg.
Então tem que ter experiência.
And we have
Esse grupo
expertise on
apresenta um
working
plano de ação de
this, and
6
we
anos.
present the
Nós
6-year action
selecionamos
plan.
isso
We select that,
então a gente usa muito o ad hoc.
and we have a lot of
E
ad hocs
esse
being
grupo, uma
used.
vez selecionado,
And this group,
chamado
once
unidade
selected,
trabalha
they
com
become an Embrapi
empresa
unit, and they work with the
precisa vir
company.
aqui, ela
The
vai
company
criar
doesn't need to come here. They will be
Então, logo,
there
a unidade é credenciada. Isso dá
as
uma
long as
velocidade impressionante
there is
ao-
the accreditation. So
sistema.
it gives a lot of speed to the system.
Ou seja, não fazer um chamado de
So we won't be called companies, as in most cases it happens. So at any time, the company will have a business unit in the project with the subject, and the project belongs to the company, not the group. The group is selected by qualification,
Portanto,
but also the project of the company. Therefore, the intellectual property belongs to the company. The patent in the academy is probably in the shelf. Among other reasons, due to the lack of culture that we have in the country. Other than that, companies are putting money there, and if they put money, we have to put ours. And anyway, we are twice monitored in the follow-up of the management contract from people from the ministries. People go there twice a year to ask for our targets, and that's why we request the targets of the unit and each project every month, although it's just a few. So this is the MCT Connect here, working with the ministries, and now the Ministry of Health, the last one. So we are a social organization, we are not a governmental body. Again, as we mentioned here, we have the Valley of Death, and we work in the TRL 326. We don't support the basic research. We don't support equipment. We don't support events unless they are events with the companies, and it makes a huge difference. So we work from TRL 3 to 6, sometimes 7. So this is our focus. And what is the system? The system is a network of technological research centers, labs highly qualified, focusing on strategies, and if you want to be a candidate, you need to be focused, and therefore this group is selected. So each unit is selected by a public call for bid. We do it once a year based on the competence of the group. It's not 1 or 2 researchers. Our average in universities would be 15, 20, in some centers 50 or even more researchers in a certain subject. And so they sign the management contract for 6 years, and then they receive the resources to operate in the project with the company. So our goal is to support development in the companies, and that's why they are called for the project with the units. It's not only us. We have a lot of LTOs, so we have technological organizations, and we have many. And our main sponsor, let's say, or our inspiration is from Hofer from Germany, but we have Denmark, United States, a lot of new people in similar cases as Embrapic entering into operation, and we have collaboration with many of them. We support 1/3 of each project, not refundable, for the development of the project with the company. And they are based on these key words. Agility, like the contract between the company and the unit lasts longer than days or even a month. We are worried because it has to be fast. And flexibility, we have the original action plan that is not a closed document. No, it's flexible. We can mix it whenever we need to make adaptations between us and the company, and zero bureaucracy. And this is something important and quite difficult to reach in Brazil. I can give you the examples later if you want to. And the companies discuss directly with Embrapi units, and they know. I don't need to mention that. We have 42 units selected from 2014 on, and we have excellent centers with highly trained people. Many of the units, as Rodrigo showed, are Embrapi units. It is important, the role of FINEP, CNPq, CAPES, and FAPES to build this structure. So now we make use of it. By the way, the 3 initial models that we had was one pilot with 3 institutions, one from São Paulo. It's a state, a public organization. IPT, so IPT in São Paulo, IPT in Rio, one in Venezuela, and Cimatec in Bahia. It worked well. It was an agreement between the CNI and FINEP. It worked well. We kept going, and now we have 42 units. The funding model is this: 1/3 of each project by Embrapi, and the other 2/3 divided between the Embrapi unit, not financed. This is what was financed before in equipment and machinery and workforce infrastructure and know-how. And know-how use as well. So at least 1/3 of the companies. Okay, she gave me 5 minutes. We have 42 units, São Paulo 10, Santa Catarina is a success with the 6, Minas 6, and we have some interesting examples with a lot of things in small cities of high success, like Santel in Guaí, and this one here in Paraíba, in Campina Grande. We have a lot of things in small cities like São Carlos and so on. And the areas they operate, let me skip it. We have specific areas, obviously working a lot in digital area, in agro-T and TICs. This is what we are today, 42 units, 17 institutes, private institutes with no profit goals. We have universities, 6 of them are federal, 4 are private, 1 is PUC in Rio. Therefore, the universities are the minority in this scheme, and we wouldn't hope for that, but the universities have a lot of difficulties in following the development of the private institutes, and 11 of them are in the Senai system. In 2015, we had 10 companies, and now we have 500. So 2016, 17, 18, 500 companies. Here we had 12, 13 projects, and here 700.
259 are over 146 patents for the companies. And here we have 15 million and now 1,200 last week. So in the period of the highest crisis in Brazil, we could break this paradigm. People said it's impossible to make the company interact with the research groups. No, that's here. It's the opposite. And today, that's how we grow. So, to summarize, that's it. We have half of this money coming from companies invested there in the profit of each one of the units, not in Embrapi, it's invested in the unit. This is how they've been growing, the companies and the projects. And the division over the years, 33% belongs to us, 18, 19, 20 from the units, that is not real money, cash, and 47, 48, and now reaching, because the oil is something huge, and it's going to more than 60% of the company. 16 oil companies work in Embrapi, only one is Brazilian. And what are the companies? I will show a little bit in details here, because you will miss something. You miss the pharmaceutical industry. Now we can go one more. However, we have Siemens, the powerful Siemens with 8 projects in equipment and hospital equipment. And here we have cosmetics, 4 competitors, Natura, Tereskin, Yama, with a joint project. It's quite cheap. Each one using their technology, and we have many other examples. Partnerships. We have BNDES, FINEP. Why? Because we are complementary in the work of these bodies. And banks. These banks support part of the company. Some of them are the subsidiaries, as Rodrigo mentioned. Desenvolve São Paulo is somewhere here in FINEP with less than 10 million. Other banks, which are CNPq CAPS, Sebrae.
Here, startups, Embrapi, of the Embrapi unit. The chance of this not working is very small, because that group is surrounded by highly qualified and focused people. And since our products can be used, they can be tested at the unit, so this is a model we use. We are negotiating with FAPESP, it was one of the first ones, and many others. We have a variety of other partnerships here. INPI, to fast track the projects and patents for models and so on and so forth. International partnerships for operational models. United Kingdom, Fraunhofer, Israel. We had an agreement with Israel, more than 20 companies. So many joint projects. And what about the companies? Here we have Petrobras. It's not one project. If it hadn't worked, we wouldn't continue this partnership. Embraer, and so on and so forth. So these are a variety of partnerships with the same companies. Here is an example of a unit of ours in Campina Grande. So they made 4 million before they entered, and now they're at 73 million. So they came from here to here. And that is valid for all of our 42 units. So our model is a model that researchers will come talk to us. Well, we don't research the people themselves. We look at the projects that they can develop as a company. And that is our message. I have brought hundreds of examples of projects. I can't show them to you right now. 12% are in the health area. That's why the Ministry of Health got into this as well. We are negotiating now with culture, energy, and others. This is the size of the resources in the health areas. When I speak of health, it's just medical equipment, hospital equipment, with the exception of cosmetics. And then here we have the TRL levels. And I think that's what I wanted to show you. There are so many other examples. Thank you. Now we would like to call Paula Salomão, Manager of Strategy and New Businesses of Antero Resource Management. Good afternoon to everyone. First of all, I'd like to say hello to my workshop colleagues. Thank Fiocruz for inviting me to speak. I think I'm going to open up this talk about investment funds, private resources. We talked about FINEP, Embrapi, the International Fund. Now I'm going to talk a bit about how this is working here in Brazil. I am an administrator at Antero. This is one of the pioneers in venture capital, which is called early stage here in Brazil, which is the investment of this first capital that enters, the first resource of private investors that the business looks for when it's starting, when it's not looking for public funds anymore and it's looking for its own investment. So it's the first venture capital fund, and that's the conversation they have with us. Antero was founded in 2005, and in 2007 we started operating a BNDES fund, Criatec 1, developed at that time. It was the first fund that had the objective of investing in these innovative startups in Brazil. And it's a fund that brought us many lessons learned. It was very successful. I'm going to talk a bit more about Criatec. It's our last year of our de-investment period, so the fund is ending, but we've had Criatec 2 and 3, so it's a model that has worked. And in recent studies of looking at our portfolio, we see that it has been a very successful initiative. So it shows that it's very promising. It's a promising fund. There are innovative startups in Brazil. At that time, if they would ask us if these existed here, and yes, they do. We have a small team, but it's very multidisciplinary, and we try to work in collaboration with our investors and the projects we want to invest in, so we are very hands-on. And in total we have done 46 investments, 6 companies in the portfolio among the ones that grew the most between 2013 and 2014, and 28 exits at Criatec. Now we have 8 companies in our portfolio. So this is a fund that is getting towards its end. So a little bit about it. It was a 100 million real fund, BNDES and BNB. It invested in a variety of areas. Here I have a little list of the health area, human health area, to talk about lessons learned, which is very important in the operation of the fund. We know what to do and we know what not to do. That's a bit of what I wanted to share with you today. As I said, we had 1,814 proposals received by the fund. This shows a lack of resources at the time for this kind of investment, and the existence of these innovative startups, these small innovative companies. The fund invested in companies with a revenue of 0 to 16 million, and this was around a number of states. They were all national investments. Speaking about our portfolio of human health, the risk investor looks at success cases. We have very interesting companies here that are still part of our portfolio, and we also have some failures that made us learn lessons that I will share with you. I think that one of the first experiences we had in this area of research was that Criatec, since it was one of the first funds in this area, we invested a lot in businesses that were beginning. There are a lot of resources for developing prototypes. Nowadays, we look at things that are in a more advanced stage, but Criatec invested in businesses that were just beginning. And one of the lessons we learned is that it is very difficult for the entrepreneur or researcher, because in the beginning they are more of a researcher than an entrepreneur, to get out of this role of the researcher and understand they need to make their business grow and be an entrepreneur. So some businesses were not successful because when you had to make this change from the researcher, the specialist, or the best researcher in this area, when you had to say, I need to leave this and look at the business and be a manager, this is a very difficult step to take. It's very complex, and we have brilliant researchers who are excellent in their areas. They are references, and they cannot take this step to the side and be an entrepreneur. So this was one of the first lessons learned with our Criatec portfolio. Another lesson learned is that it's not necessarily what is tested in the lab that can be viable, financially viable in an industrial scale. A lot of times technologies are tested in the lab, and when I am thinking about producing this in an industrial scale, it is not financially viable. So this happened with some projects. They were working very well in a small scale production, and when this started to grow, they could not keep up with this growth. So think that the fund, it's going to start with you and wants you to grow 10, 20, 30 times, 100 times. We don't want a business that is just going to double in size. We want a company that's going to increase its revenue by 10, multiply it by 50, by 100. So this leap, we need to follow this leap, and it needs to be a production leap as well. So some cases had this difficulty. And another very complex situation, which was an issue with some of these portfolio businesses, were the regulations. This case is not even here because it was in the agri-tech area, but it was a business that took 8 years to get its credentials. We were with it for 8 years. It got the correct credentials it needed, and then it was sold to an international company. So we were waiting with them for a long time. They were waiting for Anvisa, for all the regulatory agencies, and this is an issue that really affects businesses. So today we are paying attention to this. We understand some sectors in human health, it's very complex. Agriculture and animal health are not so complex as human health in terms of regulations, but this is something we pay close attention to when we're working with businesses in this area. And one last point that I think is important to talk about regarding these companies, well, actually it's two. How you get into the market. Some companies had great products that were very innovative, but the market was not ready to purchase this product. And we had the opposite case. We had a very innovative business that was a pioneer in the market, leader in their market, and everything was working out very well, like MagnaMed, which is a company of portable pulmonary respirators. It was the first company in the world to produce this kind of equipment. It's a leader and was able to use the regulations to put its product in many countries. It's very, very successful, and it was a leader in the market, and it got this product market fit. So you have this product work in the market. It's time to buy that product. And then, of course, creating partnerships. We have success cases with Vida, with Temed, another company in the medical area in our portfolio that monitors hospital beds, has a strong partnership with White Martins. So this gives the companies potential. We have Radiopharmos, and it's inside hospitals. So this network and this partnership that is established makes the company leverage itself. And this is something we learned with the Createch portfolio, and we pay attention to this when we're going to evaluate the companies that get to us with Primatech. This is a second fund. It invests in companies, in incubator companies, and we have FINEP, BNDES, Bandis, BNDNG, and it is a 100 million fund as well. Createch is in the last year of de-investment, so we're selling the portfolios. We're kind of cleaning up this portfolio, and we're looking at our jewels now to make good sales. Primatech is in the last year of investment, so it's still looking at businesses. So we invest in these businesses of 0 to 16 million reals. The challenge is to find good businesses closer to 16 million reals than to 0. And in the beginning, we would invest more in these businesses that were just starting. Now we don't have much time for this anymore, so we're looking more at this side than that side. And this is a big challenge in Brazil, to find medium innovative companies that have not received investment yet, that have potential for growth, and that are looking to find new resources. That's one of our biggest challenges for the fund. It works with a network of incubators and parks in all of Brazil. We have Mileus, which is a company in the area of biotechnology that works with food safety. It's from Belo Horizonte. You will hear from them later. It's a great company that works with transforming the researcher into an entrepreneur. And we have good expectations regarding this portfolio. So
it is much more linked to the 4.0 industry, despite investing in sustainability and creativity. I think it is related because this is a kind of business that we have this scale culture much easier when the fund is trying to scale something. We are trying to find businesses that we call a curve of revenue that is not linked to the costs. In our investment in Createch portfolio, we have a lot of legal investment. For example, in biotechnology, and you increase the revenue, but you also increase the costs, and the business is not profitable, or at least not as much as it should. So we are focused on business in scale with this potential. So having this business perception and this strategic perception of how I make my business reach this scale is much more important than talking to the investor. This fund is open. It will be a pleasure talking to you and having another opportunities in the second round of presentations that will start now. And just to finish my speech, I would like to talk about our process. I don't know if you know how to get a fund. So two things are trend in Brazil. That is being an entrepreneur or having money, because everybody wants to be an entrepreneur and all investors want to capture money. So, but there is a problem because this is a very expensive money, you know, this is difficult. So the best money is the client money, and I think this is under agreement here. So
If you have your business with a certain characteristic and you need strategic resource to help you have this growth leap, the investor may have more than money. So this kind of discussion is very welcome by the entrepreneur. I know not everybody should deal with venture capital. Not all business may have investment with this kind of investment, but it's very good for some kind of businesses. I brought here what we want from the company and how our process works. We have an identification of opportunities. We work a lot with Primatec, and we go to the states, the incubators, the parks, and strategic partners to look at the business and see what is installed there. And we talk to entrepreneurs with the potential for development, innovative development. Many businesses are essentially in a technological basis, and then we have a deep analysis of the business, trying to understand the dynamics, who is the entrepreneur, who is the business, and what is the business model, what is the stage it is, and how our money and team will help this person grow. We have a very honest relation with our potential investees, and it has brought a lot of new businesses to us. I mean, this close relation with the entrepreneurs. So we have a negotiation stage, and this is not Shark Tank, what you see on the TV. We obviously want the best return, but we have a negotiation in which we try to be fair. We have investments, we have the acceleration process that is taking care of the business. When we have it in the portfolio, we approach governance, good practices, the market, and partnerships, and strategic look. I mean, we work a lot, and then we sell, and then we have the investment. So this is the investment cycle. And what do we analyze? Well, this product market fit is how the product is in the market, and if the market is ready or not for that. We try our best to minimize the risks, and we look at everything. We have a due diligence of the business that is quite with a lot of criteria, because we have public investors. This is our responsibility to deal with the resources we have. We analyze the scale a lot, because if there is the potential of detaching the revenue from the costs, it's good, and the team is something that is emotional. I mean, it's looking at the entrepreneur and see that the team is complete and capable of working well. And if we are really having opportunities, because we sometimes have fantastic products, but it's only for small markets or niches, and we want big markets. We want to solve big problems, because the work to have the product is the same. So we focus on the big market, and processes that are quite well structured, validated product, things that are proven viable and that are well structured. So essentially we look at these areas and check if they are in line with our fund, and if they are having their heart on that. So we have resources to invest. So if you want to talk at the second moment, I'm open to questions and chat with you. Thank you.
Na sequência, vamos chamar
Now we will have Euler Santos, Director of Fundepar.
Boa tarde, pessoal.
Good afternoon, everybody. Thank you for the space to present our work, to present Fundepar.
Pelas introduções aqui do
Thank you for the
assunto, vai facilitar
introductions on
muito
the subject. It
minha
will make
apresentação
our
aqui agora.
presentation
E o
much
que a Paula
easier.
falou, né, que o
And as
Criatec foi
Paula
um aprendizado
mentioned, that
grande para
Criatec was a
Antera.
great learning
Eu
for
acho que Criatec foi
Antera.
aprendizado
I
para a
think
indústria como um
Criatec
todo, né?
was
Assim, eu vou
good
até
learning
trazer
for the whole
os dois
industry. I'll
slides de
bring
aprendizado.
two
Assim, eu acho
slides on
que
learning.
é o primeiro grande
I think this is the first
fundo, né,
main national
para
fund
investir em startups
to
que
invest
tenham
in
a inovação e a
startups with
tecnologia
innovation and technology
como diferencial
as
competitivo,
their
isso é uma
competitive
novidade para o
differential,
Brasil,
with
isso é uma
something new
oportunidade
to
grande
Brazil,
para
with
empreendedores
a
que
great
vêm de
opportunity
pesquisa,
for
né?
entrepreneurs
E
coming
empreendedores
from
que vêm de
research and
tecnologia.
those coming from
Primeiro,
technology.
eu queria fazer uma pergunta aqui, assim, para que a gente...
First, I would like to make a question.
A gente precisa de dinheiro no estágio inicial
Why do we need
negócios?
money in
Quem tá
the early
empreendendo aqui?
stage of
Alguém que fala que tá
business?
empreendendo
Do we
aqui.
have
Pra que
anyone
vocês precisam
being
de
an
dinheiro?
entrepreneur here? So let me see. Why
Tá
do you need
empreendendo, tá?
money?
Para que você precisa de dinheiro?
You are doing that, right? Why do you need money?
Para marketing.
For marketing
Desenvolvimento estratégico do negócio,
and
né?
strategic development
O produto
of the
tá prontinho
business.
já.
Okay, the
Já tá
product
pronto
is
pro
ready,
mercado.
right? Ready to go to the
Não
market.
senti muita firmeza.
Yeah, I don't see
Quase
you
pronto,
that
né? O
strong.
produto
Okay,
tá
it's
quase
almost
pronto.
ready.
Então, assim, essa é a primeira pergunta,
this
eu
is
acho que a
the
gente tem
first
que se fazer na hora que
question that we
for
have
buscar
to
dinheiro.
ask
A
when we
primeira
need
coisa é
to
por que eu
search
preciso
for
desse
money.
dinheiro, né? Eu acho
This
que essa é uma
is,
pergunta
you
bem
need to know
interessante para
why
olhar.
you need the money.
Se eu olho aqui,
If I
normalmente
look
a minha
here,
resposta ela
generally
é
my answer
sempre essa
would
aqui,
be
ó,
this
porque
one
existe um
here,
negócio que se chama
because
fluxo de
there
caixa,
is something called
que
cash
no
flow,
começo do meu
that in the
negócio,
beginning of
independente
my
de que negócio
business,
esse, se é uma
regardless of
padaria,
what
se é um
business it is,
supermercado,
if it's a
se é
bakery
uma
or a
indústria de
supermarket
base, se é
or a
uma
base
indústria de base
industry or a
tecnológica,
technological
ele vai
base
mais
industry,
consumir dinheiro
it will
do que
spend a
receber
lot of
dinheiro,
money,
né? Então,
much more than
se eu pego
receive, earn money in
negócio
the
baseado em
beginning.
ciência,
So you,
eu tenho
if I get a business based in
etapa de consumo
science,
de
I
dinheiro maior
have a
ainda, né? Eu tenho
step
uma etapa aqui
of
de
money
P&D,
consumption that
eu
is
tenho a
quite
etapa
huge.
depois
I
de
have the
transferência de tecnologia,
R&D,
seja
and
comprar
then I
tecnologia
have
de
technology
seja
transfer. I have
compor
to
essa
buy technology
tecnologia
from a
com
center
outras
or
tecnologias que
compose
estão no
it with
mercado
other
para dar uma
technologies on the
robustez
market to make
no
it
meu
more
negócio.
robust
E aí
in my
eu
business.
faço o seguinte, aí eu vou lançar o meu
And
produto,
then
eu
I
preciso de
launch
dinheiro
the
para
product,
marketing,
and I need
né? Eu
money
preciso de dinheiro
for
para desenvolver canais de
marketing and to have
ou
the
seja, é
distribution
trabalho
channels.
danado enquanto eu estou só consumindo dinheiro.
So this
e
is
aí
hard work with a lot of money spent, and then we have the valley of death where everything may go wrong.
Começo a gerar alguma
So
receita.
here I start
Até
generating
lá,
some
olha o tanto de
revenue.
dinheiro que eu já
Up to
consumi
this
pra
point,
poder
I
chegar
only
pra aquela minha
spend
pesquisa,
a
que é a
lot
minha
of
tecnologia lá
money
na frente,
to
né?
have the research
Então,
on
acho que a
my
primeira pergunta é
technology.
essa: para que eu preciso do dinheiro?
So the
Onde é
first
que eu vou
question
gastar?
is, why do I
E a
need money?
segunda
Where
é,
will I
quem é que
spend
tem
it?
esse
And the
dinheiro que eu
second
preciso
question
para
is, who
complementar,
has the
tá?
money I
Acho que
need?
o segundo
This
ponto
is
de
the second
entendimento era
point
nessa
of
linha.
understanding.
Que é isso aqui,
In this
olha.
sense, that is
Se
this
a gente olha os
shown is
estágios
this
de
graph
desenvolvimento
here.
de
If you look at the
negócio,
development
você tem
stage
lá,
of a
eu tive uma
business,
ideia, gerei
you
conceito,
have the point of
depois eu
idea,
lancei
generating the
essa ideia no
concept, and
mercado,
then
enquanto uma
bringing
empresa,
the idea
aí eu
to
fiz
the
ela
market,
crescer, e
finding
aí
a
eu fiz ela
company,
maturar, ela tá no
growing
mercado
and
maduro, né?
maturing the idea, and
Naquela
entering
linha
the
lá, ó,
market.
aqui eu tô
And in
começando a
this
gerar
line
receita.
here,
Se
I
eu
start
chegar
generating
num negócio que eu tô começando a
profit
gerar
and
receita e
revenue.
quiser
If I
colocar na
start
bolsa de
generating
valores, eu
revenue
vou
and I
conseguir?
want to put it on the stock
No
market,
Brasil não,
will
né?
it be
Fora
possible?
talvez
Well,
sim,
in
mas
Brazil,
no Brasil
no,
eu
but
não
in
conseguir ter esse
abroad
tipo
probably yes,
de
but in
abordagem.
Brazil I won't have
Então,
this
para
kind of
cada
approach.
estágio de desenvolvimento do
So for
negócio,
each
que
stage
está
of
ligado
development
dois
of the
conceitos, três
business, we
conceitos, que é o
have
de vendas,
2
a
and 3
necessidade de
concepts
recursos,
like
e principalmente o
sales,
risco. Eu
investment,
tenho
and
diferentes
risk.
tipos de investidores,
We have
né? Então, eu
different
tenho
types of
investidor lá
investors. So
lá
we
no
have an
começo, que são os
investor in the
grants, que a
beginning,
gente
that
fala
are the grants
bastante,
that
né?
we
FINEP tem
mentioned a
trabalho
lot,
muito grande nessa
and
linha.
FINEP works a
Tem
lot in
os
this sense.
FFFs,
Then
né,
we
que,
have the
que
FFFs,
são os,
that are
os,
the
family,
friends,
and fans. Eu já não uso mais o
and
FUS, né? Assim,
fans.
agora eu gosto
Okay,
mais
I
dos
don't
fans,
use the fools. I like the
que são as
fans.
pessoas que adoram o
That
seu
are the
negócio.
ones who adore your
Tem os
business.
anjos, que são pessoas
We have
que
the angels
entendem
that
do
are the
mercado,
people
não
who
necessariamente são
understand the
seus
market
amigos,
and not
mas
necessarily
entendem do
your
mercado.
friends, but they
Tem o
understand the
seed capital,
market.
que começa a entrar
Then
os
we
fundos
have the seed
estruturados,
capital
igual
with the
Antera,
structured
igual
funds
a
like
Fundepar
Antera
e
and
Science
Fundepar
estão
and Seed
trabalhando.
for Science,
Vem os
how they
venture
are
capitals
working.
e os
Then we have
public
the
offers.
venture
Então,
captors and the
eu acho
public
que essas duas
capital.
perguntas
So
é
both
importante
questions
entender
are important
antes
to
de
be
conversar com os
understood
fundos,
before
né, assim.
talking
E aí a
to the
terceira
funds.
mensagem,
And
antes
then
de
the
me
third
apresentar, né,
message
falar do
before
fundo,
introducing
rapidinho,
myself.
Eu queria só falar o seguinte.
I would just like to say that
o que é um fundo, tá? E essa figura
what
que eu
is
roubei
a
lá do
fund?
Criatec,
And
tá?
I took this picture from
que é um fundo,
Criatec, but
O fundo
a
é
fund is an agreement between investors and shareholders and the managers who are the one working on the capital and searching for the companies and helping develop the companies and then selling the companies for that to generate long-term value. So in the case of Seed for Science Fund, Fundepar is the manager. So Seed for Science and Fundepar are the managers. We went there to look for subscriptions and investments and say, invest on us, we have this mandate and this needs for investment. Would you agree investing on us? And then we have BDMG, Bands, Codegerm, Funarb, Fundep, Capemig, and Fundepar agreed, and they are here, they won't let me lie. So this agreement is what we deals and guides all the operation rules from now on. So I went to the institutions, I said I want to invest in business that are for science and coming from labs and universities or research centers that somehow connect to the labs to generate innovative businesses. I showed that to them and they said, well, I think you can do that. And lastly, the fund has this timeline. Our fund lasts 10 years. This is the average. After raising the money, you start prospecting the businesses. So this is the expectation. If we start talking about investments, this period lasts from 3 months to 1 year. So this is a long term. This is the time for us to create the building trust, this mutual trust and this unilateral relation. No, I'm sorry, bilateral trust and knowing us for real. This is like our dating time. Till the end of the fund, I have 10 years to leave the business. So I'll be your partners, but I have to share, to sell my share, because I have to give the money back to the shareholders. So this is very important because they say, then you want to sell my whole company. Well, this may be the case. So if you want to have your own company, fund is not a good partners to deal. So just for you to have this kind of understanding. And then what is Fundepar? Now I'll talk about Fundepar, what we are. We are a subsidiary of Fundep, an integral subsidiary of the center, and Fundep is a foundation for the development of research. If you don't know it, I think everybody here knows, but if there is someone from industry here, probably you don't. Fundep manages resources in research that are collected for the project. They are captured with the FINEP, BNDES, or FAPEMIG, FAPESP, or the research institutions. Fundep is one of the main in Brazil, one of the 3 big ones in Brazil. It was born to deal with FDMG, and we work with a lot of institutions in Brazil. We managed last year about 600 million reais and more than 3,500 projects. We have about 3 billion reais in research, investments, and management for the next 5 years. So this is a relevant participation in research. And Fundepar was created to go on with this mission of participation in business in research. So Fundepar is a management holding. We invested before Skid for Science in business and direct participation in business, investing about 500, 700,000 reais, and now we invest 1 million. So for example, we have an example, Detected Diagnosis. There is a company that we invested in finishing the development of products and registering the product in Anvisa and now marketing. And as Paula mentioned, it is in the portfolio, and it is a development company and for DNA diagnosis and safety. So these are business examples related to health and biotechnology.
We created an investment fund, and we also invested in some change structuring businesses. So I'm going to speak about this during 3 slides about Biotech Town and Seed for Science, that are the investment vehicles we have today for innovative businesses in science and biotechnology and life sciences more specifically. The Food for Science has the proposal of being the first institutional vehicle for investment in the fund, the FIP, the investment fund with participation. We want to develop entrepreneurial culture in centers and universities, prepare this company for growth, and introduce the cooperative culture to these researchers and entrepreneurs. We learned with Paula that not always a researcher will be an entrepreneur. They need to participate in some way, but maybe we can't complement these skills. So we need to find these complementary skills for the entrepreneurship to bring this forward. We invest 1 million reals in this company in the first round of investment. So this speaks a bit about the limitations we will have with some businesses. Maybe, let's say I need $40 million for a clinical trial, so we probably will not be the fund that will participate in this, but we could compose a part or a group of investors. We also understand that we need to bring expertise into these management decisions, management support, bring organizational concepts. We're going to create a business. We also help a lot for looking for other fundraising. So those companies that we invested in directly, for each real we invested, we brought in another 9 from private investors and public investors, helping the networking to develop businesses, sales. In our businesses, we developed all of them. We invested in all of them when they had no revenue, and for example, 3 years later, we have these businesses making 14 million reals with more than 160 employees, highly qualified labor, people with master's degrees and PhDs. So all of the partners either have a master's degree or PhD. So these are highly qualified people, highly qualified labor that we are developing in this market. We also have some competences and internal accesses to help with the development of products. For example, we have great possibilities with DDC and other units to have this vision of how do I get this product, develop it, use this technology to turn it into a product and put it into the market. We have looked at businesses in bio, nanotechnology, advanced materials, and data sciences. These are the more traditional science areas in Brazil. Also applied to bigger markets in Brazil like the agribusiness, health, well-being, industry, retail as well. But business in retail, not commerce itself, the business to business. And when we are going to invest, Paula talked a little bit about this. Obviously, we look at the team, how committed the team is, what is this team lacking, and can we help in some way? Because if we can't, it will be difficult for us to start this partnership. The business will not grow in the way it should. We look at the market. Sometimes you have great technology, it solves a very relevant problem, but the market is too small. We've seen this happen a lot in diagnostics. It's very small. How do we connect this to a bigger market? Because otherwise we can't justify the investment in our participation. We help to build the business model. Obviously, the entrepreneur already knows about this. We feel safer about it, and our technology is a competitive differential in the market. For example, I joke about Uber, Airbnb are great businesses that we could never invest in because technology is not involved here. It's not the big benefit here. This is a critical point for us. This is the agreement that we made with our investors. So finally, we need the investment fit. My 1 million needs to be able to make a difference and take this to the next level so I can get more investments. If anyone is interested, we can talk later or check out our website, fundepar.com.br/seed4science. Here you can sign up. The fund has existed for 6 months. We have 4 more years of investments. We can work on a lot of different opportunities for investing. The second vehicle of investment we have, I'm going to be fast about this. It's for startups. It's Biotech Town. It's a partnership of Condengio with Fundepar. And we created this to think about biotechnology. It's private and to offer businesses and products to the market. So it covers lots of services in biotech and life sciences. And it's located in Nova Lima, 25 kilometers from Belo Horizonte. It's a technological center. It's a new area that's being developed in an area called Alphaville in Belo Horizonte. It has 3 business units. This is a map. You have the business developing area. We invest in the startups and the idea stage. We become partners. Put these resources there up to 150,000 reals to help structure these ideas and look for partnerships. Then we have an open lab that is under construction. You can rent this space to develop products. It's an open lab and it has the intention of making these connections with other labs that have already an established infrastructure, like for example a partnership with CT Vacinas. They have the structures there for vaccinations at UFMG, and I can create partnerships to use those labs over there by the companies that are working in our facilities. So we want to create this network. And then we have the CMO. It's under construction as well. It will be the first place where you can construct your biotech products instead of having to mount your own structures. So this comes from when we started investing in companies like Detecta for diagnostic kits, and we thought, wow, if I'm going to invest in 20 of these diagnostic kit companies, I'm going to have to build these locations, and if I have this shared structure, the investments are more viable. So for example, we have the business development program. Some companies have already been invested on. Oncotag, I think it's from Unicampo. It's FUNED. Oncotag has diagnostic kits for ovarian cancer. You have RIA, that is from Unicamp. It's also diagnostic kits for a variety of human and animal diseases. You have medical devices. You have different components, different biological components, so you can have this structure. Another case that biotech is supporting are the international connections, soft landing for international companies. Urba Mannheim is a company that is establishing itself there and looking for partnerships to develop its competences in the national market. This is an Indian company. And we have the Inside program that are companies that establish themselves there to develop their partnerships. The partnerships between these biotechnological companies is already installed there. We have some consultancy companies as well. 2 new contracts were defined. We have one with a business developer in the health area. And another lab is also there with an innovation workplace. So our doors are open if you'd like to get to know our spaces and develop biotech businesses. Here's a nice photograph. Well, I think that's it. That's what I wanted to get across to you. So you can see what kind of money you're looking for, and on our end, what alternatives for investments and business models we have provided. And later on, I'm open for us to speak more about this. Here's my contact information. So now we will call up Eduardo Henrique Soares, Director President of Biominas.
Boa tarde a todos. Prazer estar aqui.
Good afternoon to everyone. It's a pleasure to be here. The members of our table, we are all partners. It's great to be here. And I was going to joke and say I'm last because I'm the only one who's not talking about money. But we just heard from the king of money, so now it's not funny anymore. I'm going to speak a bit about what we do in general terms. We work before what everybody was speaking about here. Our main focus is to identify the sciences, the quality sciences of a project, a high quality project in universities or in the entrepreneur's head, or that is still in research development phase, and give the support for this project to advance in the chain of development and eventually receive investment or financing by some of the agents who act in the market. We have various cases here with Fundepar, with Antera, and other partnership with FINEP, BNDES, as well as companies that are using the benefits of the Embrapi. As Jorge said, related to an institution that receives the resources and conducts its development. So we are a private institution. We've been active for 29 years, and we're focused on creating businesses and in creating businesses in life sciences in the broader spectrum involving all areas of knowledge in bio. I used to joke and say that life sciences is everything that is not IT, but we are now IT, so it's not funny anymore. So that's it. We've been active for 29 years. We've been able to do a lot of things during that time. Some of the things we do is analyze technology. We have analyzed 3,000 technologies during these 29 years. So we know the main universities and research centers very well. We know the researchers and understand where these projects that we looked at are. Since 2015, we created a pre-acceleration program for startups in the life sciences area. We created this because we were a bit jealous of the pre-acceleration programs that existed for IT. So we decided to create the pre-acceleration program for bio. And people thought this was impossible because it's a different process, etc. The investments are different. But we decided to do it anyway, and we got support from Sebrae and created a methodology. So since 2015, we have conducted 8 rounds of this program, received more than 1,000 projects, selected about 150, 160 that passed on to this pre-acceleration program. Some turned into startups, and then the growth process continues.
Historically, many of the people know us as a company incubator. We've never been a company model. We are a manager of company incubators. They're in Belo Horizonte. So since 1997, we incubate companies. It's been 23 years since we started doing that. And during this period, we graduated about 35 companies, and we have other 14 or 15 that are in our building currently. And this group of companies over the last 20 years has reached more than 2 million reais, and now their income is about 10 million reais. It is also important to mention that we've generated more than 200 million reais in taxes, which is also a contribution for government. Since 2001, we've been studying the biotech sector, and in our studies they refer to the sectors, and we have some main studies like, okay, I don't know if they are the main because they are the main studies or because nobody else studies that, only us, but you can trust the data. And we end up being the information generator on the sector, which also helps us understand the status of the companies and understand what they are doing and in what stage of maturation each one of the businesses are. Since 2014, we are partners with Bayer, which is a big association of companies in the world, and we have an event here every September called Bio Latin America that I invite you all to be part of it. You can enroll now, and we are working very well since last September. FINEP, BNDES, and Embrapi are invited to be our sponsors. I hope you are our sponsors. And other companies who are here, please, you are invited to that. And we are associated to Interpharma, that is an association of pharmaceutical companies in research. So a good part of the international pharmaceutical companies are part of Interpharma, and we are the only non-pharmaceutical institution being part of Interpharma, and we are very proud of being a member of Interpharma. And we work on a lot of innovation programs with them, especially this acceleration program that we had twice in São Paulo in partnership with Interpharma. As we are in an investment session, just to bring a little bit of our history, we've already been investors, like putting money on stuff like a program we have with the International Development Bank, and we received some money to work in companies and invest in companies, and our quote partner that was not a quote, but they were a fund from BID, from the Development Bank, and we have some investment. Two successful cases that we had was of a company called Prodimol, which created and developed diagnosis kits, and this company ended up being sold to Inverness, that is an American group, and we sold by 3.5 of what we've invested. And another success case that we had was Alvos, a company that we also sold to Ouro Fino. And as you know, they deal with animals. And the interesting thing in this case is that they work on a technology developed by Fiocruz that was a molecule called CN14, and CN14 was a candidate for the Leishmaniose vaccine, and Dr. Miriam Tendler was conducting the study, and what we did was to create a company. We created this company called Alvos with a science and technology offered to the company, and we financed Fiocruz to this company, and we could invest. We put money on that. We brought another investor that was Fundotec 1 from FITEPTO, and I'll talk a little bit about it later on. And along with that, we could have money from FINEP, and this made the product possible to be developed, and it called the attention of Ouro Fino. Ouro Fino then bought not the technology, but the whole company. They took the whole company along with the project.
Isso o projeto está em fase 3, né?
It is a phase 3 in development of the human vaccine, phase 3 in Africa, and now we are in contact with Emilia. So this is a successful case. So 2 cases, obviously I only brought the successful ones. I won't bring the ones that we wasted money, and we have about 8 of this. 8 that we invested, 3 were good, we recovered the investments, and 1 we are still partners, I'll show how it is, and the other ones we literally lost everything, and it is part of the history of investments. Yeah, statistics are good, so that's why we try to focus on the positive cases. But it is always complicated, you know, life of investors. You think that we are always earning money, but it's not that simple. Biominas, as I mentioned a little bit yesterday, we are divided into 4 pillars today. It is part of our rearrangement that we did last year. So we have the academy area, because we are trying to understand the perception, the problems of the country, and we lack education and entrepreneurship in education. So we took the role of generating knowledge and innovation and spreading these teachings so that researchers and entrepreneurs and even professionals in the area, they can understand this process of entrepreneurship. So we are launching some programs. Some of them will be long-distance teaching, and next month we'll be launching these courses. We keep on going with the think tank initiatives. We are in partnership with Deloitte, and we'll launch this study in the next Bio Latin America and in September. And I have only 5 minutes to speak. And corporate that we work along with big companies in this effort of innovation. So we are trying to convince more and more companies. And this is George's work to bring companies to be more innovative. And I can see, George, that there is no pharmaceutical company, either Brazilian or foreign company, in our portfolio, and we need to increase the participation of the pharmaceutical companies in this area. And the 4th area is a little bit of our core, that is the identification of ideas and projects and help projects in all bases. And we have this line of development that goes from projects that are under research, then we go through the pre-acceleration program and then the acceleration program, and we also have incubators that in the final phase of development they start the production, and we have our building where the companies may be allocated and then develop. Regarding investments, we have 2 initiatives here just for you to have an idea. I know you've seen it a lot today. Everybody has shown this graph, so I won't describe each one of the items, but just for you to understand where we work. We work basically here, so before being a company, when it is still an idea, we help structure the business model. We help creating the logistics of the business, And then we start giving support with acceleration programs in which we end up being partners of the business. So we have the participation, the shareholder participation in the project, even though we don't put money on that, and trying to bring investors and big companies to support investments and to support funding. So we have many projects that are in this process being invested by some of the funds that are on the market. This is, for example, the case of Mileus. It is incubated in our building, and Antera and Primatec and also Fundepar invested on that. So, in this sense, with the portfolio of companies that are the ones we have share participation, we have 8 companies. Linhagem, that is the last company in our investment program, along with the Development Bank, working with animal genetics. And then we have BCAM producing biodiesel from kitchen oil. This is a very interesting product. And this is something that went through Embrapi. We got resources from them because we have a partnership with Votorantim, and Votorantim then took the project to be supported in Embrapi. We tested engines with biodiesel in Cimatec in Salvador, and the tests were excellent. We were really proud because we tested biodiesel in 10, 20, and 50%, and it worked very well. We are quite satisfied with our results. Hakuna, that develops food from insects.
Vet Solutions, que desenvolve vacina para leishmaniose.
Vet Solutions with vaccines for leishmaniose, and Nanopec developing a film to protect the life of fruits. Duna with bioinformatics services. Adarna, that is in the formation and management of the health sector. And Epitamer, that produces treatment for cancer, and they are under an acceleration program in London. And lastly, just to go back to this slide, we are also supporting a fund in Minas Gerais, that is this fund here, that FIR Capital manages, and Codege is one of the main investors in this fund. We are in the last phase of collecting resources to operate. It is a fund that is from the end of seed and starting of venture capture, so we can create a chain of investments that start with our initial programs, go through the Fundepar programs and so on, and until the end where we have more resources. So we are very pleased to see everything going on. So this fund starts with about 100 million, may reach 300 million. The rules are basically the same. We have this perspective of investing in 5 to 7 companies with more resources. And just very briefly, the last slide that I will talk a little bit about FIR and why FIR is capable of managing the funds, because probably one of the main biotechnology and health successful cases in Brazil is Biobras in the production of insulin, and then it was sold to Novo Nordisk with another company, Reaching, that is Beyond, and now they are creating a factory in Belo Horizonte to start producing insulin in Brazil, which is not done since 2001. And another successful case of the company is Oncoclínicas, that is the biggest cancer clinics in Brazil. They had an investment in the beginning, the business grew, and now they have an income of almost 500 million a year, and they are giant in the health sector. And without mentioning their investments in other areas, not necessarily bio, but nanotechnology, with this institute, CESAM Brazil, which created the first spin-off that is SANIL, and if you go outside, there is a tree where you can, a pole where you can recharge cell phones with energy generated from photovoltaic cells, and they pass through the system developed by SANIL, again with the support of Embrapi. So that's it, my time is over. It was enough, so invest with BNDES and beyond and so on and on. So that's it, thank you very much.
Pessoal, e para finalizar, vamos chamar João Paulo Pieroni.
The industrial complex of health services in BNDES. Good afternoon to everyone. I wanted to thank Fiocruz's invitation for me to be here. It's great to be here speaking to entrepreneurs who are interested in technology and to speak to these important partners like FINEP, like Biominas, people from venture capital, so it's a great pleasure for me. As you can see, BNDES has a great relationship with most of our partners, especially regarding the health system. We're very active. We've been active for more than 15 years. Today I'm speaking about this department that deals with all of the health projects in BNDES, whether it's industrial, medical equipment, diagnostic equipment, or just health services in general. And we have this relationship with the venture capital team as well, relating to health. So basically I will speak I think it froze here. I'm going to speak quickly about the two main subjects, a bit of BNDES's vision on the health market, the pharmaceutical market, as well as, okay, here we are. And a bit about what BNDES's role is, the products that are available, new initiatives that the bank is working with. So just quickly, the pharmaceutical market in Brazil, despite our deep recession that we've been living through since 2014, it's a very resilient market, so it is still growing, and the perspective is more growth above the global average. It's the 7th market in the world. It will become the 5th market, so we have chronic diseases, the population is growing, and we have structural characteristics that make our market very resilient, and it grows despite the complicated economic scenario in Brazil. And beyond that, if you have seen the presentations, you know this slide very well. It is a summary of what BNDES's vision is of the pharmaceutical industry in Brazil and its evolution since 2000, and what is the space that we can see for this development. So the industry has gone through a process. I'm speaking about Brazilian companies basically. We have gone through a process of improving quality, good practices of manufacturing. We are internationally accepted today, and we started with the production of the national production in 2000, which was very relevant. First they were non-qualified copies, now they are qualified. The current stage today is incremental innovation. I think Rodrigo can speak on FINEP's behalf how we see projects by big pharmaceutical companies migrating from these copies to medications that have a different dosage, a different combination that is based on the existing products. And the big challenge is to create new products, new molecules developed in Brazil, molecules, let's say, that speak Portuguese. And the results that are frustrating that Jorge spoke about, which is how the Brazilian industry doesn't have much relationship with the universities, which are the places that have potential for new products. So we are at a stage that we have not taken this step yet. So a second aspect of biotechnology here is the effort that has been done since 2012, 2013 in this search for plants that produce biotechnology, as well as in the GMP mode, good practices, the challenge of thinking about making these projects sustainable. So this is the summary of our vision here. We've tried working on this at BNDES and trying to push this innovation trajectory towards technology. For this, we need an ecosystem that has been developed. No country in the world can deal with this innovation if it just comes directly from the pharmaceutical industry. So we have tried to look for partnerships with different elements in this chain, and the relevant part is here. We have the regulatory aspect, which is totally relevant in this industry, whether it is intellectual property or other aspects. In Brazil, we are not used to investing in radical innovation, so we have a lot of resources, but not a lot that will invest in risks. So we have funds, but they're still very small. BNDES is trying to discuss how to share this risk in a large scale and apply this to innovative projects, small and large companies, startups, and how to involve a chain of services, hospitals, universities. Interesting movement is when they go abroad looking for technologies, whether they are not available in Brazil, or to accelerate their processes. So a lot of businesses have opened spaces abroad in the US, in Europe, or Canada. Of course, we don't want this to just be limited in this sense, but in the innovative industry we need to be present outside of Brazil. And the movement, well, we hope it will exist. We hope to attract multinational companies to Brazil, to look at Brazil as a platform for exporting. Right now there are a lot of plants that are closing in Brazil, unfortunately, because they are old production plants and they are less feasible nowadays. But we are looking at clinical research, and I think Brazil has in general improved the regulatory environment for new trials. So that's a bit of the scenario. And how does BNDES fit into this, or try to induce this scenario? So here, the Brazilians here already know the bank, but basically we are 67 years. We are being very attacked right now, and honestly, I think it's unfair in most cases. 67 years of activity, a public bank, 100% public, and it is the main long-term instrument in Brazil, and recently, during history, its role has changed. First it was more industrial, and now it's more modern, and it supports medium companies, sustainability, and all of these actions. BNDES, in contrast to some other discussions, if we compare it to banks in other countries, we can see the differences between other developmental banks like KfW in Germany, JFC from Japan, the Chinese bank. These are regional banks. I'm not comparing it to the World Bank or multilateral banks. So today BNDES is the third or fourth biggest developmental bank in the world. It has great performance rates. Many times it has good return rates as well compared to other development banks. Here the planning, the strategic planning of the bank, which was finalized last year. Maybe for those of you who haven't looked at this so closely, we're going through a transition that's very relevant in our history, because we lost a competitivity tool which had a lower cost than the banking practices in Brazil. So today we have migrated since last year from TJLP to TLP, which is based on inflation, and BNDES is closer to the current market conditions. This imposes a number of challenges: how to be more efficient, how to be faster, but also how to maintain the development role. That is why we have restructured our strategic planning, and this is summarized in this slide. Infrastructure, the bank is still important, it's relevant. We have more than 20 years in big projects in Brazil. The productive structure, and for the first time, highlighting here education, health, and security, and our transversal missions that cover all projects: sustainability, innovation, development, and capital development. So health is at the top of our strategic priorities right now. And how do we perceive our role? I think those of you who know our activity know that the bank never sees itself just as a financing agent that analyzes the project and provides the resources. We try to have a more strategic vision to create these discussions on public policies with other agents, and that is where the development comes from, and therefore the investments. The vision is not so much about waiting for projects and actions, but more advancing. That's what we always believe makes us more efficient. And how does BNDES act? Compared to institutions that are more dedicated to other stages, the bank supports all growth stages of a business, from the seed money way in the beginning, like Criatec and other initiatives, up until the grants, which have been reduced recently. I'll speak a bit about this, but we have some, and of course the cooperative credit for mature businesses, like our partnership with Ouro Fino, which Eduardo mentioned. Ouro Fino, we had shares at Ouro Fino, and then we took the business to IPO, and its participation was reduced. Today it's a business that's open in capital markets. This is the bank's role. We work in all of these stages. In the case of health, today we have a portfolio of more than 200 projects supported in the health area, $3.5 billion in health. And today, these are being managed by our department. In biotechnology, we have about 1 billion reals in different areas, 22 contracts. All of them are available online. All BNDES contracts are available online with information about clients, rates, guarantees, objectives, and our biggest objective is to try to reduce these gaps in the value chain of biotechnology. This is very difficult because the bank is not involved with discovery or research. There are other agents involved here. But from the scale up, the bank is involved with projects in public institutions like Fiocruz and Butantan, also private institutions. Not many, but it's already relevant support, about 1 billion reals, part of them in partnership with FINEP.
Agora travou mais uma vez. O próximo detalhe de cada uma
I'll go into details of each one of them. So I'll divide basically in three big actions. First, traditional financing, credit. I won't go into taxes and interest and so on. So the bank has finance lines which make it possible to finance almost all kinds of investments, may it be for construction or improvement of a factory, support of the idea of the company with personnel and equipment. Participation goes up to 80% of the project, and we have here a maximum of 20 years. And with this change in the fines, BNDES is quite clear, has a quite clear and limited role in finance above 10 years. So less than 10 years, we have a credit market in Brazil that somehow works poorly, but works, and BNDES is more focused in projects demanding a mature time that is longer, like 10 to 20 years. In investment funds, BNDES is the main player in Brazil with more than 3 billion reais and more than 43 funds, and Criatec is one of them, and there are other 42. And we have a fund dedicated to health called FinHealth that is there, and it's there because we have a period of de-investment, so we are trying to work with a new fund dedicated to health, and we couldn't put that on the market yet, but BNDES is the main investor in investment funds, and especially in the stage of L stage with Criatec or the Angel Fund. That should be the first initiative that BNDES, if they have the angel investor in the startup, they can have the same amount of investments in the startup. It is a project that is ongoing. And lastly, we have some projects. The bank has more than 25 projects of non-refundable resources that are resources from the bank itself, BNDES, that were guided for technological development and for the social fund and cultural fund. And here I brought some examples, only 2 examples in fact, of projects supported like Butantan and Fiocruz. And in particular with Butantan we have huge support with dengue vaccines that today is in phase 3, and we have this expectation of submit Anvisa in the last semester with the first vaccine of dengue in Brazil, second in the world. So the first one was not effective. So probably this vaccine has the potential of, in fact, being a product developed in Brazil with the potential to be a world product. And even part of this product is licensed for the mature market. And in Fiocruz, we have a series of projects that go from neglected diseases to diagnosis, and we got the support from Henrique Pena Center in prototypes. The bank supported the pilot itself, that is a pilot plant for biotechnology that is filling the gap that we always see in Brazil, a gap that we wanted to build, and it will be used by Fiocruz and also used by the whole market, and Priscilla will speak better about it. And what is more interesting to say here, I'm just finishing, 1 minute. I would just like to mention 2 or 3 initiatives. The first one, bank has recently launched a call for studies for the Internet of Things and applied to health. In fact, it applied to smart cities and environment and cultural. That was the identification of the biggest potential. There was an Internet study carried out by McKinsey, sponsored by BNDES. So we have more than 6 projects in health, and we selected the best ones. We are analyzed these 6, and this is a very interesting action, and also the potential of scaling for the unified health system and the private sector relevant in application of technology of IoT in health. And the second and more relevant to you who are entrepreneurs or have your startups are the program called BNDES Garage. You can go to the website and go there, and in the first stage you have the module of creation and acceleration of startups. We had 120 startups selected in a building that is here in Rio, in the space of WeWork. And the second stage, I was talking to Eduardo about the creation of an innovation center of BNDES here in Rio to be managed by a third party. We are having the manager selected and the space as well. It will be 3 kilometers away from BNDES, so it will be from the Port to Gloria, somewhere in between, and will be a big space that BNDES will be the main sponsors, and the cost of installing a startup or having big companies with a lower cost. And the idea is that we have about 8,000 meters squared. It's a relevant space, and we'll have about 20 million reais a year to maintain this innovation center of the bank. This is still under selection. Probably next month we'll have the result of who will be the manager and all the beginning of the project that is quite nice, and a good close relation of the bank with the startups, okay? So lastly, this is my last slide, and I always look at the scenario today and see a lot of difficulties, and if we look at it as a film, we at least have a relatively optimistic view of what we faced, what the pharmaceutical industry or the health industry faced over the last 15 years in a scenario of regulatory uncertainty. We didn't even have a regulatory agency that was strong, and companies based only commercial aspects. So we now have a lot of strength. We are acknowledged. We can work with the companies, and Blue shows that the pharmaceutical sector is the one that invests the most. A lot in-house yet. That's why data is relevant, and we hope this industry to be an innovative industry so that we can catch the opportunity of biotechnology in a competitive way. I mean, not only a biosimilar industry to produce to Brazil, but in the end, we need to think about the unified health system and the access to patients, okay? So that's it. Thank you.
Vamos fazer breve
I'll have a brief closure here because we are a little bit late, so I would like to thank the speakers, all of you long-time partners of Fiocruz or with the potential to be partners, because in your organizations you have the idea of fomenting technological innovation and bringing solutions for public health, and I bring here a little thought for you to develop when we talk on behalf of Fiocruz and we think about the access models to capital and funding for public institutions of public right. We have a lot of restrictions. So Fiocruz has been discussing a lot internally, trying to make use of the science and technology landmark and other discussions to reinvent the models of access to capital and the business models to generate technological innovation, and also corroborating with what Kabir mentioned. I think in this sense, and especially in public health, we need to gather efforts from the public and private sectors on behalf of the national and international public health, that many times we are also gathering financing for neglected diseases that are not of interest of all players in this chain, and we need to deal with that. So I have a lot of institutions here being part of this process of new public policies for us to try to reach new solutions and new ideas, just like Embrapi has done, and with an innovative model of innovation for companies, as you mentioned, so that we can find alternative ways to build this model. So how can we do that? And João showed $3 billion in the Department of BNDES in the industrial complex of health, but when we look at the refundable resources, it's just a small part of the global value. So we have this challenge. I hope in the next panel we may start some of these interactions. I will invite you all to take 10 minutes for a coffee break. We have a coffee for the speakers who didn't have lunch, and we'll take this 10 minutes to reorganize the room. If you were not here in the beginning, we'll have each one of the speakers on one table with a person from Fiocruz, so that the researchers of Fiocruz and others present here may talk and establish their first relationships. Thank you all.
session.
Oh, he has a lot of business cards.
Yes, I've been collecting them this week.
Opa, thank you. Thank you.
O que vocês vão fazer com os cartões?
Vai fazer sorteio.
Ah, uau!
Terá um sorteio ao final. Conseguiu? Ah, você já botou seu papelzinho aqui, né?
Já botei.
Tá. Não sei. Surpresa.
Eu queria...
Desculpa incomodar, é que a gente está pegando os cartõezinhos de visita, que a gente vai fazer um sorteio ao final da sessão. Aí a ideia é a gente poder saber, tem até uma...
entendi. Eu
tem um
não sei.
registro. É só o... Você tá com o cartão? Tá querendo concorrer? Não. Tá.
Amanhã vai ter 5, né?
Já peguei. Cartão de visita? A gente vai fazer um sorteio ao final da sessão?
Pensei que era para fazer doação, aí eu já estava preocupado aqui, mas se for só isso...
Opa.
Jogado por 2, né?
Aumenta a chance. Opa! O Eduardo ia botar um monte aqui. Você tem cartão de visita?
O que está sendo agora?
Não, então, é uma sessão que a gente vai fazer de bioconexões. Opa, valeu.
Tem que pôr o cartão?
Isso.
Eu assumi uma nova área na Fundação Ezequiel Dias tem um mês, eu não tenho ainda. Tem como participar do outro jeito?
Pode ser um papelzinho com os contatos.
Vou anotar.
Você tem cartão de visita? Oi? Cartão de visita? Eu não sei se eu vou ficar porque tem uma palestra para vocês. Vocês vão participar da sessão agora? Então pode vir se caminhando. Vocês têm cartão de visita? Tem um papel. Pode fazer um papelzinho depois e deixar aqui. Avisa elas também, por favor.
E o Manguinhos bota também cartão?
Ah, tá botando. Opa. Tá botando. Só tô guardando o pessoal entrar, porque o pessoal tá ganhando. Vou ter que montar aqui. Não, vou... Aí eu vou falar pra vocês, não, pra vocês fazerem duplas. Eu só tô deixando quem vai participar pra botar pra dentro. Vamos ver, vocês vão rodar. Vai fazer uma... A ideia de dinâmica é que vocês passem 2 minutos com cada um.
Ah, com cada um.
Isso, é rápido. 2 minutos. Aí, só essas primeiras 20 minutos a gente faz assim, aí depois aí abre para livre. Pode. Não, nos primeiros 20 minutos você pode participar, mas aí você não necessariamente vai encontrar com ele. Pode acontecer de encontrar, pode acontecer de não encontrar com ele. Pode acontecer de você encontrar com ele ou não, porque como é randomizado, eu vou fazer duplas. Vai ser agora? Pois é, porque tem um monte de gente aqui, eu tenho que chamar a pessoa para dentro aqui. Pessoal, com licença, quem vai participar da sessão? Eu preciso para a gente poder começar a sessão, senão eu não vou conseguir. Aí, pode, por gentileza. Para a gente poder começar a sessão. Sim.
É interessante botar também o telefone?
Sim, sim, pode pôr.
Você não botou?
Não, mas aí não, se botou, tem e-mail?
Não botou.
Se botar, poder botar e-mail, telefone. Vocês vão participar da sessão? Não, acho que não. Tudo bem. Com licença, Helena, vocês vão participar da sessão? Você vai participar? Não, você vai conversar com startups ou com os investidores. Sobre algum projeto. Não, algum projeto que você tenha, se for um investidor, algum projeto que você tenha uma demanda de investimento, se é uma startup, se ela tiver alguma tecnologia que te interesse. Você oferece para que ela tenha algum, pode ter algum, não sei. Então, a startup é mais eles trazendo ideia para a gente. O investidor é você demandar investimentos deles, que você possa viabilizar algum tipo de se casar, se não casar, por isso que é 2 minutos de encontro só, porque você pode não casar nada, 2 minutos não interessa, passou rapidinho, vai pro próximo. Pessoal, boa tarde. Boa tarde, pessoal, mais uma vez. Desculpa interromper as conversas de vocês. A gente vai abrir esse primeiro momento, a gente vai dar 4h20 agora.
Okay, at this first moment, like from 4:20 to 4:40, we'll have a forced interaction, a controlled interaction with you. Then we open for interactions as it is happening now, because the idea of this session is that you interact with the more people you can, because basically you interact with one person and you miss opportunities. So I would like you to work in pairs. You may take the pairs that you are working now, so if you don't have a pair, find it here, get your places.
É porque eu ia ter os cartõezinhos de... Tá, tudo bem. Então, enquanto sou duplo. Então, ele
so, who's your pair? Okay, yeah, it's randomized.
Você fala com ele,
so you talk to him.
Isso.
Yeah, you talk to him.
Vocês já se conhecem, não?
Do you know each other?
Então pode ser assim, vocês são uma pessoa só. Tá? Vou considerar vocês uma pessoa só. Já botaram lá, né? Podem botar depois. Então, achem sua dupla, tá aí. Acho que eles dois ainda vão entregar mais dois, porque só entregaram só. Vou botar mais dois aqui. Ali atrás? Ali. Oi? Então, você quer falar só com ela? Então acha tudo duplo.
Nós já vamos sair.
Pois é, pessoal, vocês já, eu preciso fazer acontecer, rodar aqui. Então, já que vocês já estão aqui, ela entra agora para uma interação e depois a gente pode até voltar com ela, mas só para. Aí busquem seu duplo. De repente ela está sem duplo, fala com ela. Vocês com ela aqui. Isso. Aí você interage com elas duas aqui. Com elas duas. Aqui. É, isso, cola nelas, todo duplo de vocês ali. Pessoal, todo mundo com suas duplas, fazer o seguinte.
Hey guys, everybody with your pairs, let's do the following. For the interaction, just a second, people.
Quem consegue, porque senão não vai conseguir organizar esse nível, esquece. O que é importante é garantir que todo mundo consiga falar, entendeu? Então, se eles estiverem monitorando aqui, porque assim, essa lógica de todo mundo rodar... Pode, entendeu? Estimula, vê assim, elas querem falar com quem? Entendeu? Porque não tem tanta gente querendo falar com tanta gente, então tem que garantir que com quem elas têm que falar a gente consegue, entendeu? Elas querem falar o quê? Com eles?
Tá, vou fazer, vou mudar um pouquinho de novo. Tá, pessoal, rapidinho, só um momento de silêncio rapidinho, só pra gente definir as interações.
Just a second, let's make some silence here first to define
por favor, por
the
gentileza,
please.
Quem tiver interesse, você já se aproxima, fica das áreas, divulga um pouquinho a dinâmica. Se sinalizem com quais os lugares que vocês querem conversar, vocês já se posicionam.
Check
e a gente só
who
faz a
you want
interação
to
nas
talk
mesinhas
to and
com
get
as
your
pessoas que estão.
positions and get
Tá,
your
para garantir que todo
interactions with
mundo
the
consiga
people who
falar
are there,
com
so
aquele
that
que tá,
everybody can talk to those who want
então,
to.
agora essa dinâmica está valendo.
So
Obrigado.
this is the dynamics that we have now.
Esse pendrive aqui, eu achei que fosse do Aziz, aquele gringo, né? Aí eu perguntei, ele falou que não é dele, eu não sei quem é, e aí tem uma opção de trabalho dentro deles e não tem nenhum ali que a gente relaciona com a apresentação, até difícil, vai tentar achar isso aqui.
ele faz todas essas coisas. Eu
E aí,
Pessoal, desculpa incomodar de novo, interromper, mas é por uma boa causa. A gente vai fazer sorteio, a Gisele vai sortear aqui o cartãozinho, e a gente vai estar sorteando 5 livros, né, do nosso homenageado do evento, que é o Dr. Reinaldo. Então, a Gisele vai tirar os números, vai tirar os nomes e a gente vai... Vamos lá? Milena Magalhães. Parabéns. Opa. A próxima pessoa. Opa. O Kabir não tá. Kabir saiu, então perdeu. Próximo nome. Quem é? Beatriz Fiado. Ó, essa é uma marmelada, hein? Marmelada. Tá, peraí, ela foi, ó, Beatriz foi muito gentil, ela vai deixar eu sortear novamente, porque ela tem um autografado pelo autor, então, ainda, dá licença, Fernando Porto, também outro marmelada também, O próximo é Paulo Afonso Nogueira, pode vir, Zebe Supremo, livro do Dr. Reinaldo, isso, pode voltar aí. Nada. E o próximo, tem mais 2 livros, Oscar Araújo. Oscar. Parabéns, Oscar. E o último nome, vai para o Eduardo da Biominas. Livro do nosso homenageado Dr. Reinaldo. Nada. Então, pessoal, agradeço a todos pela presença. Isso não é de maneira nenhuma querendo despedir, tchau, mas podem ficar à vontade, conversar. Muito obrigado, pessoal, pela presença.
Beleza. Eu quero depois